Okay, I think we're ready to start. First of all, I want to welcome everybody to this symposium. This is our second symposium as part of the celebration of the 50th anniversary of the CS Mott Center. It has been a really a very exciting year with a lot of activities, and we will be completed or ending in next June 14 of 2024. As I mentioned, we have a symposium last month, this symposium on pregnancy, and we have another symposium coming early February. So I hope to see many of you also at that symposium. I want to go quickly. First of all, before I start with a quick review about the Mott Center, I want to thank some people. First of all, Dr. J. Ramadoss, who has been leading the organization of the symposium. It has been an incredible work. Congratulations. Also, the people who are behind the scenes working on this. You will see around George, who will be in charge of all the computer systems, all the technology. Of course, Alisa, who is around coordinating and looking that everything moves smoothly. And Sharon Pepper, who is organizing behind the scenes also and coordinating that everything is on place. So thank you so much for all your work. All the symposiums have been successful, and this one also will be. Thank you. This type of events cannot happen also without the support of representatives. I want to thank Natera, Rapid FN, and BioLegend. We like to suggest that during the breaks, please take time to visit the booths, talk to them, get a lot of information. They're very helpful, and some of the information will be also helpful to you. So what are we celebrating? We're celebrating the 50 years, as I say, of the CS Mott Center, who was established in 1973. This building is really unique. It has almost 24,000 square feet of laboratory space in 3 floors. And in the first floor, we have also the administrative space, as well as a bioinformatic core and the clinical research center, which I'll talk to them in a minute. It was established, as I say, in June 14, 1973, as the center of the Department of Obstetrics and Gynecology. In 1983, it was established as a university center. In 1988, was designed as an entity of the School of Medicine. The dedication of this building, who was in June 1973, was done in the presence of Ruth Mott, and you can see also her picture there. This is the wife of Mr. C.S. Mott, who donated the funds in order to build this facility that you see now. This is Mrs. Ruth Mott. That is some historical pictures of that time, with the first director of the center, that was Dr. Tommy Evans. Again, here is Ruth and her husband at that time. It's interesting, the originally design for this building was 7 floors. It has only 3 floors. It seems the money was not enough to build the other 3 floors. But we're happy with the floors that we have so far. And again, this is the same picture that you see in this room of Mrs. and Mr. C.S. Mott. Our mission of the Mott Center was to promote personalized approach to medical treatment and care that improves health outcomes for women, men, and children in Detroit and around the world. That was the mission in 1973. And I can tell you that that mission continues and is exactly what we are doing today. And I think even with great success. How is this building organized? We have, because human development encompasses multiple areas, but all of them are interconnected. But at least in terms of organization, the Mott Center is divided in these subgroups or units. We have the pregnancy and neonatology unit, the female infertility unit, the male infertility, the oncology, And of course, this group that is the Clinical Cancer Research Center. I go quickly, you will start. Oh. Okay, thank you. So the neonatology group is composed of both. In every group, you will see a mixture of MDs and PhDs that are working together. You will be meeting during the day through the presentations on many of them. The female fertility group are focusing in oocyte biology, endometrial biology, uterine biology, myometrium biology. Again, it's a group of fantastic PhDs as well as MDs. The male fertility focuses, of course, the sperm biology, and the oncology group associated with research in gynecologic oncology. Not only the research, but also clinical trials. And that's what leads us to one of the gems of the Mott Center, that is the Clinical Research Center. At the present time, the Clinical Research Center has 7 coordinators. Those coordinators have 20 years of experience of running clinical trials. They can do everything from the first contact, IRBs, recruitments, and the running of the clinical trial. So this was a quick summary of what is the Mott Center. I can say that our portfolio in terms of grants continues growing, and our ranking within NIH also continues increasing. I don't want to spend too much time on this. Again, I want to welcome you, all of them, to this meeting, and we will enjoy the science.
Thank you. Really appreciate all of you being here. Thank you, Dr. Moore, for the welcome introduction today for the Pregnancy Symposium. It's been an amazing time last 2 months trying to prepare for this symposium. I'd like to especially thank the keynote speakers and all the external speakers we have today. And let's get started on the first presentation for this morning. So the United States Surgeon General has labeled the use of electronic cigarettes as widespread. There is an unfounded presumption that the use of e-cigs is somewhat safer than traditional tobacco cigarette smoking, which leads to the use of e-cigs during pregnancy and exposing the pregnant patients and the fetus to many potentially harmful chemicals. Despite the limited knowledge about the safety that these e-cigarettes pose to the public, we have about 27.5% of the teens and about 28% of the young adults who use e-cigarettes. The current generation of e-cigarettes use a handheld battery, which in turn rapidly is connected to a metal coil, which rapidly heats and aerosolizes the e-cigarette liquid. And what does this e-cigarette liquid in turn contain? It contains 3 parts. One is called the VGPG. Okay, this is a combo juice called as a vegetable glycerine and propylene glycol. Depending on how you mix this VG and PG, it gives different kinds of user preference profiles. For example, if you have a high amount of PG, like let's say 80% of this combo is PG, then it gives the user a throat hit feeling, a throat hit sensation. But in contrast, if you have like 80% of PG, then it gives you a huge cloud. You know, some people would like to see a huge cloud. Then you make your e-cig, custom make your e-cig such that you have more of this PG. And then the second component is, of course, you have the nicotine, which is common with the traditional tobacco cigarettes. And there is a multitude of flavorings. So the bulk of the talk today, we need to have this VGPG because that is the base for the e-cigarettes. And then we also have, we use the nicotine, but towards the end, I will also show you some of the work that we are doing with flavorings and what flavorings in turn do to pregnancy, both from the maternal and the fetal perspective. So we custom built and engineered a vaping system that comprises of a software interface that controls the timing and the electrical potential output supplied to an atomizer. So there is a programmable atomizer that is integrated to a commercial tank to generate the e-cigarette aerosols. So we built a custom inhalation chamber to house the pregnant rats and a continuous air compressor combined with an activated charcoal filter that safely removes all these e-cigarette aerosols out of the vaping system. The bottom left panel here shows that the US Surgeon General report on e-cig nicotine concentrations that is being used in the humans. So we try to recapitulate what is reported in the US Surgeon General report, and we used a similar concentration of the nicotine in our vaping paradigm and exposed our pregnant rats to 1.5 hours each day during the pregnancy. They get exposed a couple of days every week throughout the pregnancy. And then we utilized mass spectrometry to identify what aerosols are there in the vaping chamber. And here we can see a number of aerosols that we have listed from our mass spec. And the size of these letters is proportional to the abundance of these chemicals inside the vaping chamber. The most prominent ones would be, of course, nicotine, glycerol, and propylene glycol. But then you have many other organic compounds. These are all breakout compounds. So when you heat this e-cigarette aerosols, they break down and create a number of organic compounds, which are all found in the vaping chamber. So this is our model. So we use 3 treatment groups. One is a pair-fed control, which is pair-fed to the nicotine group. And then we have an e-cigarette base group, which contains VG and PG, vegetable glycerine and propylene glycol base. And this is a second control group. And then there's a third group here, which contains the base e-cig, the VG-PG. On top of it, it contains the nicotine, which I had just shown you in the previous. Right now, we are not adding the flavorings, but towards the end, I'll show you what is it that we find with the flavorings. The nicotine dosage produces serum nicotine concentrations similar to that of the human smokers, moderate human smokers. And so in the first study that I'm going to show you here, we hypothesized that prenatal exposure to e-cigarette aerosols with and without nicotine would significantly alter the lung genomics in the fetus, the lung structure, morphology, as well as function. So the top panel here shows that the fetal and the neonatal weight were significantly reduced in response to the presence of nicotine in the e-cig liquid. But the gross weight of the fetus was not altered when we just gave, when the rats just vaped VG-PG alone. Okay, so this is the first observation here. And the same thing with the crown rump of the fetus, it was significantly decreased only in the presence of nicotine. Just VG-PG did not do anything to the crown rump length. The bottom one shows 10 days postnatal, and these growth deficits persist beyond the last day of the vaping exposure. We stopped the vaping 2 days before parturition, but then even after 10 days, we measured the weight up to 10 days after birth, and we see that the growth deficits persist much beyond the last day of vaping during pregnancy. Okay, so something which is unique to our lab is like we do multiple organ systems for the fetus because we are basically a fetal development lab. And the first organ system that I'm going to be showing you here is the vascular system. And followed by this, I'll show you some data from the respiratory system with the lungs and nutrition. And I will end it with some in the brain, in the fetal brain development. The top panel here shows that the maternal and the fetal heart rate both were not altered in response to VG-PG or with nicotine. And then the bottom panel here shows that we measured this using Doppler ultrasound flow probe, and we found that there was a significant deficit in the urinary blood flow with the nicotine, and it trended down with the juice group, which is the base, which is VG-PG. And similarly, there will be found a decrease in the umbilical artery blood flow as well in the fetal compartment. Then we performed an RNA-seq on the urine artery, which is the main artery that supplies oxygen and nutrients to the fetal placental compartment. And this urine artery, we isolated it. We performed an RNA-seq and found a number of genes associated with the vasodilation, specifically with the nitric oxide system, significantly altered with this RNA-seq. The bottom panel here shows that, again, there was a significant decrease in the expression of the endothelial nitric oxide synthase, which is the rate-limiting enzyme producing the vasodilator nitric oxide in the primary urine artery. And then in the right panel, we use a method called as pressure arteriograph, where we recapitulate the blood flow conditions using a system ex vivo, and we measured the vessel relaxation in response to acetylcholine, a classic endothelial derived vasodilator, and found that there's a significant impairment in the arterial relaxation of the urine artery. So this tells us that so there are three things here. One, there is a decrease in the contraction of the urine artery in response to an agonist. Second, there's an impairment of the vasodilator system in the urine artery. And third, there is a decrease in the urine artery blood flow. So with that, about the hemodynamics, I will move on to the next system, which is the fetal lungs. So the fetal lungs, we performed an RNA-seq again on this, and to identify the molecular targets disrupted by prenatal e-cigarette exposure. So we examined on gestation day 21, which is like about 2 days before parturition in this species. High throughput RNA-seq identified about 23,500 genes for comparison. And the top panel here shows that about the 10 most downregulated genes, and these are the log2 fold changes. And the bottom panel shows with the e-cig base. This is with just the e-cig base, and this is with nicotine. And independent of whether nicotine is present or not, there was a significant alteration in the genes that are being altered. And we also found that when we examined the top downregulated and upregulated genes, several of these genes played regulatory roles in the development of lung cancers and many of the inflammatory conditions in the lung and associated with respiratory mechanics and respiratory morphology, lung morphology. We did an additional analysis of the amino acids by doing an HPLC from both the maternal plasma, the fetal plasma, and then we also took from the male lungs and the female lungs. A number of amino acids were altered. There was a differential expression between the male and the female lungs. And similarly, something which is very unique for us is most of these alterations are found in the male lungs, but we do not find them in the female fetal lungs. So e-cigarette seems to be affecting the fetus in a sexually dimorphic way, which the males are heavily impaired, but the female fetuses seem to be not having that many issues with the lung. So a distinct feature here, what we noted was the excitatory amino acid glutamate in the bottom panel, which is an excitatory neurotransmitter, but is also cited with pulmonary hypertension through NMD receptors in the lung. A number of amino acids were altered in the lungs in both the base as well as the nicotine group. So we then examined the lungs. The fixed lung weight and the volume were both significantly decreased in the nicotine group. So something that is very distinct is with reference to the base group, there is no alteration as far as the whole lung weight or the gross measurements are concerned. But with reference to the fine measurements, that's where we see the difference with the VGPG as well as the nicotine. So to identify the changes in the structure of the alveoli, we analyze the sections of the lung for measures called as the mean linear intercept, which is commonly used parameter to examine the free space within the lung parenchyma. And we also use this, and this MLI was significantly increased in both the e-cig base and the e-cig nicotine group. And as far as the radial alveolar count is concerned, and this was significantly decreased independent of whether nicotine is present or not upon vaping. And this indicates an increase in the size of the distal air spaces. So this combination where you have an increase in the mean linear intercept and a decrease in the radial alveolar alveolar count is demonstrates the characteristic of an emphysematous phenotype with larger and fewer distal air spaces. You can start seeing that how it trends from control towards the nicotine. So these larger and fewer distal air spaces. Then these morphological changes. So again, the top panel again shows these morphological changes with alveolar septal thickening and overall increase in vascularization, and they were accompanied by altered respiratory mechanics. So we use an equipment for this called as the FlexiVent, which is used to measure pulmonary mechanics on postnatal day 10 to determine the functional outcomes of e-cigarette exposure during pregnancy. So there are two things I want to show you here. One is the resistance. The pulmonary resistance was significantly increased whether there was nicotine or not, but e-cig vaping increases the resistance. And the right panel here shows the pressure volume loop. So this is the pressure in order to expand the lung to a certain volume. What is the alteration of the pressure, which is basically the work done. So if you see this work done, it shows that again there was a significant increase whether there was nicotine or not. And this estimates the amount of atelectasis prior to the pressure volume loop maneuver, and it was decreased in both the nicotine and the base groups. So with that much about the lungs, we will, this is a small one slide I want to show you about the flavorings. So we have now started doing different set of flavorings. So these are the base ingredients that are found in the flavoring. So there are thousands and thousands of flavor. You know, you have strawberry flavor, you know, vanilla flavor, and so on. So we cannot be taking all those flavors and doing. So what we did was we took the base ingredients which are there to make those flavors, and then the around 45% of all the e-cigarette flavors, they contain vanillin. So we took this vanillin, ethyl butyrate, ethyl maltol, methyl cyclopentanol, and menthol. And menthol is like a separate flavor. Then we mixed all these five and then subjected them to the vaping. And the rats really like the flavors. And then, and you can see that with the flavor, there was a significant increase in the growth. Okay, that was very surprising for us. But same way, this is the gestation day 20 fetal weight and the placental weight. Everything was increased with the flavors. I haven't done any more now. We have just done some basic analysis, and the right panel shows here that we did some mass spectrometry of the plasma of the serum samples after flavor exposure, and a number of metabolites were altered in the maternal plasma. So with that, I'm going to go to the last organ system that I'm going to show today, which is the brain. And so as we see here, this is we took a structure which is very closely associated with learning and memory and this is a fetal hippocampal formation. The left panel here shows the inner part of the hippocampus called as the dentate gyrus, which contains a cell type called as the granule cells. And these granule cells, when we image them, we have, this is how the control looks, and this is how the nicotine looks, and you can see that the branching and the morphology of the dendrites is significantly, there is significant more arborization of these neurons, okay? And these dentate, these granule neurons are like the input neurons in these structures. And so we wanted to find like why is this morphology altered in this way in the fetal hippocampal formation. And so in order for that, we targeted a particular pathway called as the mTOR, which is very closely associated with development, fetal. And what we found was that this mTOR, the activated form of it called the phospho-mTOR, was significantly expressed. Okay, so this is the total mTOR, which was not altered between the groups, and it's very closely associated with development. The left panel here shows the activated form of mTOR called as the phospho-mTOR, and this was significantly increased only in the presence of nicotine. And this was also found specifically in the male fetal hippocampus. We don't find these kind of alterations in the female fetal hippocampus. And then the bottom panel here shows that the specific structure in the brain where we find this mTOR being altered is also the same dentate gyrus the hippocampus has an inner seat called as the dentate gyrus and outer seat-shaped structure, which is called as the pyramidal field, composing of the output neurons called as the CA1 pyramidal neurons, CA2 and CA3 pyramidal neurons. But we specifically find this phospho-mTOR in the dentate gyrus granule neurons, and we can see that it significantly increased the phospho-mTOR in response to the nicotine. And then confirming our immunoblocks. And then downstream of this mTOR, we have specific proteins to determine whether it is an mTOR1 or an mTOR2 pathway. So this protein here, we have 4EBP1 and P706K. These two are downstream of the mTOR1 pathway, and we can find that in the presence of nicotine, there's a significant increase in this phosphorylation, as well as the total amount of these 4EBP1 and P706K. 6K, and showing that the mTOR1 pathway is significantly altered. The bottom panel is PKC alpha, and this is downstream of mTOR2, and this also seems to be altered with e-cigarette vaping. Okay, so these are some accessory proteins which come and bind to the mTOR complex. One is called the Deptor, the Raptor, and the Victor, associated with both the mTOR1 and the mTOR2 pathway. And one of them is mTOR2 and the rest are mTOR1. And Deptor, we didn't find any differences, but then with the Raptor, again, there was a significant increase in the male hippocampus in response to nicotine, and the same thing with the Victor associated with the mTOR2 pathway. Again, we have a significant increase in the presence of nicotine. And the bottom panel here shows pictorially what is happening in the fetal brain, in the male fetal brain. And we can see here that in the controls, we have the mTOR, the Raptor is increased, the phosphorylation of mTOR is increased, downstream the P706K is increased, the 4-APP1 is increased, totally showing that the mTOR1 is significantly increased in response to e-cigarette vaping at the end of pregnancy in the fetal hippocampus. And the bottom right panel here shows again that phospho-mTOR is increased, the Victor is increased, PKC alpha is downregulated, and this is a classic pattern for mTOR2 to be upregulated. So the summary of this is both mTOR1 and mTOR2, and we are now investigating whether this mTOR in turn is connected with the brain morphology changes that we showed with the hippocampal dentate gyrus granule neurons in the first slide. So the take-home points of this, there are 3 organ systems basically I showed you here. One is with neurobiological, so things that we have been working on, and are one with respect to the RNA-seq and with different pathways. Some of these, I didn't show the data today to you, but these are things that we have been working on. And then we have pulmonary data with impaired lung growth, altered lung mechanics, and inflammatory environment based on the RNA-seq. In the cardiovascular system, we have the uterine blood flow specifically seems to be decreased. It's a prime artery which has about a 30 to 50-fold increase in the blood flow in the 3rd trimester of pregnancy compared to the non-pregnant luteal stage. And that artery seems to be specifically altered, unlike the other systemic arteries in the maternal compartment. And we find there was a significant 40 to 45% decrease in the blood flow in this artery, leading to a decrease in the delivery of oxygen and nutrients to the developing fetal-placental compartment. So with that, I would like to thank all our funding sources and to our lab members, without whom none of this data is possible, Mr. Naik, Mr. Jansky, Karabuli, and Hong Jian. And our collaborators from Harvard, Dr. Steen, Dr. Riley from San Diego State, and CS Mott Center. We work with Dr. Moore, Ding, and Dr. Chen. Thank you, and I'm happy to take any questions.
Hey, a quick question. So looking again at the results at the beginning of the nicotine and the phase, it suggests that the action is not just the direct effect of the nicotine and the fetus, or is the maternal response that is affecting all those organs? Nicotine cross the placenta and reaches the fetus?
Yeah, nicotine does cross.
It does cross.
It crosses, but it could be a combination, just to answer, it could be a combination of the effects. There could be maternal effects, like how we showed that there is a decrease in the maternal uterine artery blood flow, which decreases oxygen and nutrient delivery to the fetus. So that means that there is significantly a maternal component, and there would be an additional local component, probably.
Is this reversible when you stop smoking?
I don't know the answer to that question. One thing I can tell is we stopped the vaping exposure 2 days before delivery, but then the growth deficits, we measured only till we do only pregnancy and neonatology sign. So up to like postnatal day 10 we measured, and we did not find any reversal of the growth deficit. It was the growth deficits persisted. So I don't know if it will, maybe when they are like much older, if those deficits go off. I don't know the answer to that.
If the cooling system has an effect, yeah.
Can
Some people using cooling system, is that they have any effect here?
A cooling system?
Yeah, when they fire.
Yes.
So I think his question is pretty much, I was thinking in that direction as well. It's something you might want to look at, because projecting this to the clinical realm, you eventually get to a point where you get well funded that this is going to translate to human pregnancies. One thing we're going to be asking is, how do we prevent this? And so finding what, if it's reversible, and more so drugs we use to treat people who are addicted to tobacco, like Wellbutrin or even the patch, things like that. How does the rat respond to that? That, those are things that you may want to kind of tease out before you go to the decision.
Well, these are good questions. We are actually working through some pathways right now where we can try to mechanistically block some of these things and find in our system. It's those proposals are just underway, so I just want to put those things here. But at the end, it's always important to see that we're able to translate this to in a clinical setting. So that's the point as well taken as well. So thank you. Yes.
Yes, thank you very much for the interesting talk. Can you speculate the reason for the sexual differentiation that you found in two of your models? That's number one.
Two of the organs.
Yeah. So two of them. The second question is that the same? I don't know much about smoking. I don't smoke myself. But the question is that the same in clinical realm? So in adult, for example, do we have sexual differentiation?
Yes.
In the way men and women respond to the nicotine?
It's a very good question. So this really depends on the teratogen or the exposure that there is in pregnancy. There are some exposures where there is not that much sexual dimorphism at all, and I have seen that. But with respect to nicotine, there seems to be a dimorphism even among adult smokers, something nothing to do with pregnancy at all. That there is a greater damage in the male lungs, but not in the female. And but with e-cigarettes, with cigarettes, it seems to be profound. But for every teratogen, it doesn't happen. We have personally experimented other teratogens where there is no sexual dimorphism at all. But here there is, yeah. Yes.
Just a quick question. So going back to the question that we just asked, with the vaping, is it pure nicotine as opposed to cigarettes, which you get, carbon monoxide and a whole bunch of other components as the tobacco burns? Are there differences between the two, and what effects do they have?
So I'll answer your question, and I'll complete answering yours. I didn't complete answering your question. So yes, when we do a mass spec of the smoke, there are about 1,000 substances in traditional tobacco cigarette smoke, which includes some of the substances you mentioned as well. But e-cigarettes have way less. They have only about a fraction of it, like 50 to 70, 75 aerosols in the e-cigarette, and clearly the number of aerosols is way less in the e-cigarette. But some of them are so like we can even see things like acetic acid, acetone, so many things we can see, but they're on small fractions. To what extent each of them play a role, that I don't know. And the second part of the question, which I didn't answer for you, is like, how do we, mechanistically, how do we know what in turn results in the sexual dimorphism? So we are not there yet, but we are currently doing some gene expression analysis and trying to find out if there are some pathways which are specific in different structures. Like, is there a common pathway that is a gene expression change pathway that is affected, let's say in the male placentas, the male fetal lungs and the hippocampus. So is there something common? That's something that we are investigating. And that's, I think, a very nice realm of investigation. So thank you. Stop there. Okay, thank you. And I will introduce the next speaker. So our next speaker is Dr. Ding, an assistant professor at the CS Mott Center for Human Growth and Development and the Department of Obstetrics and Gynecology here at the Wayne State University School of Medicine. And the topic is sex dimorphism of placenta and its response to exposome. Welcome, Dr. Ding. Thank you.
Thank you, Jay, for the nice introduction. So today I'm going to talk about the sex dimorphism of the placenta and its response to the exposome. So we know that placenta is formed during the pregnancy and is a very important organ as the bridge between the fetus and the mother. It has multiple functions, including for the transport of the nutrition, metabolism, and remove the waste, and the hormone production and the protection of the fetus from infection. So here I want to emphasize that during the pregnancy, the placenta is a very active immunological organ which can protect the fetus from virus or other environmental stress. So the first fundamental question we have here is, are male and the female placentas asexual? So to answer this question, we did an experiment that we get the mouse placenta from female and the male. We sent for RNA sequence to see what is the difference between the female and the male at the transcriptal level. So firstly, we have 2 days of the pregnancy, which one, the first one is E12.5, and the second one is 17.5, is near the term, and this is considered as just have the placentation. So here we can see that the origin one is the early male placentas E12.5, and then we have the female placentas E12.5 female. So as we can see at E12.5, there's a distinct difference between the gene profiles between male and the female. And then we also have the 17.5 female and the male. We see there are some difference, but there are also a huge portion overlapped in the gene profiles. So this is just showing that the top differential expressed genes between the male and the female. We see there's distinct differential expressed genes between the early placentas. So we want to look further into what is exactly the difference between the female and the male in the early placentas. So here we see that the top biological process here, there's a lot of developmental difference between the male and the female, such as the animal organ development, and then including a lot of like cell differentiation, tissue development, and epithelial development. So it is telling us that during the early pregnancy, the male and the female placenta, the developmental process are distinct differently in the placenta. And then this is the early placenta. Then we go to the late placenta, it's a 17.5. The same thing, we also analyzed the top differential genes, and we found a lot of genes are differentially expressed in the late placenta. So to confirm these findings, we confirm the pulse control, we confirm the expression of the X chromosome dependent genes, so such as XIST, which is highly expressed in the female. And then the DDX3Y is highly expressed in the Y chromosome, which is only show significant expression in the male placenta. Besides that, we also validate the expression of the X or Y chromosome independent gene, such as the LLC 7A11 and the interleukin-7 receptor. So here we validate this Y or X chromosome independent gene has significantly differentially expressed in the female and the male. So the same analysis as we did with the 12.5, we also see what is the most different biological process in the late pregnancy placenta. Then surprisingly, we found that there's a lot of biological process associated with the defense response and like the immunological immune response to, in the female and the male placenta. Because this is the male control versus female control, so the females are control. So we can see here, the male placenta seems to harbors more immunological active pathway. So to confirm that findings that we found in the mouse, we are wondering what is happening with the human placenta. So to study this one, we have the collaborator from China. They have the placenta from the selective abortion from 6 to 8 weeks, and then they perform the RNA-seq. So as we can see here, it's a distinct difference also in the human placenta between male and the female, including X chromosome and the Y chromosome genes, such as the DDX3Y we validate in the Y chromosome. So apart from the chromosome-dependent genes, we also found the chromosome-independent genes that have been differentially expressed in the male-female placenta. So this is the top biological process. As we found in the mouse, that immunological biological process has been differentially expressed in female and male. Here is the same story that in the male and the female in human placenta, there's also a very distinct difference between in terms of the immunological pathway. As we can see here, the immune system, the defense response, how they respond to the external stimuli. So then we have the early placenta, then we want to see what is the term placenta. So this is the database we have access through the public database. We get them and reanalyze the data from the database. So this is to showing the top differential expressed genes. There is some variation within the group, but overall, they have also very distinct expression of the genes as well as the biological process. So here we see that in the term placenta, some similar biological process stands out, such as the humoral immune response and how the antigen associated with the MHC class 2, and then have the acute inflammatory response here. So from this validation and the RNA-seq in the human and mouse placenta, we know that female and the male placenta has significant different gene expression profiles, and the immunological differences are present between the female and the male placenta. So why this is so important? So we want to answer the second question is, how is the male and the female placentas response to infection or the environmental stressor? As we know that when the mother is pregnant, so they have possibility to get a viral infection as the infectious stimuli, or they are exposed to the air pollution, such as, or the nutrition, or different stress. So it's called the non-infectious stimuli. So we'll activate the maternal immune activation. So this will lead to the like the local inflammation in the placenta. So this could lead to bad or good consequence in the fetus. So the good possibility is that the fetus will become more resistant to the future challenge. The other aspect is that it could lead to the susceptible of the fetus more to the asthma, autoimmune disease, or viral infection, and even cancer. So during the whole process, when the mother is exposed to the viral infection or the environmental stress, so there is a maternal immune response to these stimuli. And as well as in the placenta, there is a response in the placenta, which can be a bidirectional system that the mother affects the baby and the baby affects the mother. So it's very important for us to know that how the placenta responds to this stimulus. So firstly, we want to study the viral infection. So we choose the Zika virus that we have been a long time to studying this virus. So this RNA virus that of the flaviride family is the transmission is primarily through the bite of an infected mosquito. So if the adult get infected, they don't show much severe symptoms, maybe just like catch a cold for 2 or 7 days. But if a pregnant woman get infected with this virus, it's very dangerous. It has been shown that it can cause the severe microcephaly if some mothers are infected with the Zika virus. And the other pregnancy outcomes are linked to the miscarriage, impaired growth, eye defects, and the hearing loss in the child. So this virus is very dangerous for the pregnant woman. So here we did the experiment that we infect the pregnant mouse on E12 8.5, just around the time the placenta is forming. And then we did the RNA-seq for the 12.5 and 17.5 for the placenta RNA sequence. So here I will show the data in the 12.5. So here, the first thing we want to see here is that the 12.5 males, the blue one is the male control, and the orange one is the male Zika. So we see after Zika infection in the male placenta, there's a very a different response in terms of the genes profiles. And then same story as the female, they have a very a strong response to the viral infections. Here is just to show that this is the female control and this is a male control, the baseline difference. And after the Zika infection, the difference are still there. The female, this female Zika and this is the male Zika. So further looking into what is the unique or what's the similarity. So of course, the female and the male placenta, they share some similarity in response to the virus. So this is the common differential expressed genes in the female and the male placenta responding to the Zika virus infection. But what we are more interested is what's the unique ones. So here we look at the unique pathways in the female placenta. So here we see that a lot of the top pathways are associated with the metabolism, such as the different enzyme and different amino acid and drug cytochromosome P450. So here we can see that in the female placenta, when they respond to Zika virus, it's more like showing metabolic changes in the placenta. So what is happening in the male? So when we look into the male, as we saw in the baseline difference in the immunological difference, here we see that they have a more stronger immune response in terms of response to the Zika virus, such as the infection pathway or the neutrophil, the NETs, neutrophil extracellular trap formation, phagosome, and the NOTs like a receptor signaling pathway. And then this is showing us that the immunological changes is more dominant in the male placenta compared to the female placenta. So here we also want to see the biological process. So the one that I showed before is the pathway, and the biological process is what's more like in detail what is happening here. So we can see here that a lot of these homeostasis or the process of the metabolic has been cell adhesion, metabolic, catabolic. This is all associated with the pathway we just mentioned. The metabolic pathway has been significantly impacted in the female placenta. So here comes to the male placenta. So what is the most unique biological process in the male placenta? As we can see here, there's a lot of biological process has been affected in the male placenta, interleukin-1 beta protection, activation of the innate immune response. A lot of these associated with the immunological response. So we also confirmed the finding that So in the female placenta, when they exposed to the Zika infection, there is no significant increase of this interleukin-1 beta production. However, in the male placenta, there's a significant increase of this. This is very important finding. It's telling us that to respond to the same stimuli, female and the male placenta may produce different profile of the cytokine and the chemokine. So in the clinic, if we want to use some biomarker, such as interleukin-1 beta, it will have because if we say, okay, there is a higher expression of IL-1 beta, we predict there will be a bad outcome of the pregnancy, but we have to consider the gender or the sex of the fetus when we predict the result. Because if we didn't see IL-1 beta in the female, in the pregnant woman carrying the female baby, then we say, oh, there's no problem. But that's not true because the placenta response is different. Maybe we will be wrong in that case. So it's very important to see the response of the placenta to the stimuli. So here we summarized our finding of the mouse placenta to the Zika infections. So we see that when the pregnant mouse is stimulated with the Zika virus, in the male placenta is more immunological response to the virus. However, in the female, it's more metabolic response to the viral infections. So the second stimuli we have mentioned is the non-infectious stimuli. We studying the benzene exposure that nowadays, there's a lot of air pollution going on. The air pollution comes from a lot of different sources, such as the industry, the trash burning, and the vehicle pollution. And the benzene is one of the chemicals that rank in top of the pollutants. It's colorless and it's very highly flammable. It can evaporate into the air very quickly. So there is a study in Detroit showing that the preterm birth rate is very high in Detroit, which correlates with the air pollutant area. So where air pollution is higher in the area, there is a higher rate of preterm birth. So this is also because the pregnant women are more susceptible to the air pollutants because they have 40% increase during the pregnancy. They need more oxygen and they need to have more burden to carry their baby. They need to have more calories and burning more calories. So these increased needs for the air and make them will inhale more polluted air. So this leads to a lot of advanced pregnancy outcomes, including the preterm birth labor, stillbirth, low birth weight, preeclampsia, a lot of developmental problem in the children. So in this case, we want to study the benzene exposure during pregnancy, how they trigger the placenta response in the female and the male. So the same thing, the mice mated the next day, the plug day is 0.5, we consider embryo day 0.5, and then we expose the mice through the whole pregnancy. So here we show the data of until day 12.5. So the same thing that after benzene exposure, there is a very distinct response of the male and female placenta to the benzene exposure during pregnancy. So here, this is the orange one. We can see this is the benzene male, this is the control male, and this is the benzene female, this is the control female. This is clear to showing us there is a response there. And then this we also want to see, is there anything in common? Yes, there is the differential express gene, which are both up in the placenta, female and the male, responding to the benzene exposure. But more interestingly, what is the unique ones? So the unique ones to the male, we can see that the top ones are a lot of developmental process that we have mentioned in the baseline difference. And then this is unique to the male. And then we have the one that a lot of transcription process has been affected in the female, such as as you can see here, transcription by RNA polymerase and transcription DNA templates. There's a lot of transcription related biological process has been affected in the female. So this is the pathway analysis. So we can see here that this is, as we can see, insulin secretion, and then they have gastric acid secretion, pancreas secretion. So this is a lot of like developmental, especially the GI and the liver. This problem is telling us that in the long term, the offspring that we also know from other reports that when the mice is exposed to the benzene exposure, they may develop diabetes. So this finding in the placenta is critical because we can link the offspring phenotype to the what is happening during the placenta to find what has been the regulator, what is been the trigger for the offspring phenotype. So this is telling us that in the male placenta, as other research has found, the phenotype is only in the male. Why is only in the male? Because the male placenta has a lot of developmental problem in terms of the diabetes-related genes or pathways has been affected. So this is unique to the males. And then we have only one pathway unique to the female. So that is the virus, the herpes virus, one infection. So this looks like from the benzene exposure that the male has more complicated response to the benzene exposure. So from these, we know that this mouse placenta, when they expose to the benzene exposure, in the male is more developmental problem, and in terms of the GI system, the liver, or the other metabolic, or the other developmental process. And then in the female, there's a lot of transcriptions, because transcription is very general and complicated terms. Transcription or alteration can lead to the synthesis and the translation of the proteins, the difference. So in the female, there will be more to investigate into the transcription pathways. So here I wanna summarize that the sexual dimorphism of the placenta, that we know there's an intrinsic difference are there, and then they also have a differential response to the stimuli, and then the response to the stimuli, depending on the stimuli, whether it's virus or it's environmental stressor like the air pollution, is completely different to the fetus. Every fetus is very unique to responding to the same stimuli, and also the sex dimorphism of the placenta plays an important role in this immunological response. So that's all for my presentation. So thanks to the NIH grant and thanks to Dr. Moore's lab, and then our collaborator in China, Dr. Ai Hua Liao. So I would like to take any questions.
One comment that there are lots of single cell data sets right now that you want to refine a little bit more to see what cell types generate those differences between men and females. Probably some of the public resources, I think some that we generated here at Wayne State could be used maybe to further refine where it is coming from. And then once you have those signatures specific to those cell types, maybe you can try to deconvolute a little bit your both data and to find out a little bit more how that plays in.
Yeah, thank you for the suggestion. We are working on the deconvolution of the analysis of different cell types because we also want to know why it's coming from. It's from trophoblast cells or is the macrophage also have the sex dimorphic. It's a very interesting area. Thank you.
As a clinician, I can't help but always think how this applies to, a real life scenario. As you probably know, female fetuses tend to do better than male fetuses when they're born, preterm, which is why we have ways to one of the prognostic factors, one of the biggest prognostic factors for survival at preterm is the sex of the baby. How does this play into that? Because from what I'm hearing, female fetal tend to do well with respect to metabolics, but males tend to do well with respect to immune issues. Is there a link between your data and that finding we see?
Yeah, that's a very good question. So I also think exactly the what you are thinking. So how to explain, trying to explain the sex dimorphism in the preterm birth. So I'm thinking that because the female and the male trophoblast cells, they are responding to the different stimuli. So we know that viral infection and the bacterial infection can induce a preterm birth, right? But how the female and the male placenta, specifically trophoblast cells, how they're responding to the stimuli. In the male placenta, they may secrete a huge number of cytokines, like interleukin-1 beta, as I was showing there, which can just trigger the preterm birth, induce the delivery, early delivery. So, but female placenta has a kind of like not very much huge like the cytokine storm. So they keep them alive. So the male baby cannot survive because of the cytokine storm happening during pregnancy and leads to the preterm birth. But the female placenta, because they are controlling it better than the male placenta, that's why they protect the baby, the female can be born. So, but that's my hypothesis. I didn't see a lot of researcher in terms of finding the cause of the sex dimorphism in the preterm birth. But I do very interested in that area to do more experiment to see that.
Quick follow-up. Thanks for that answer. Most of the things you showed were genetically, were differences in your genes. Do you think that hormones might be playing some role?
I think so.
High testosterone, high estrogen.
I think so. Yes, because hormones has been related to inflammation and then the control of inflammation. So I think the hormone definitely plays a role. And a lot of researchers, when they study sex dimorphism, they focus a lot on the hormone. So in my study, I want to kind of study more on the side of the gene or immunological changes and kind of dependent of the hormone, because this is an area that's under investigated.
Sure.
So I want to study that. Thank you.
Thank you.
Yeah.
And just as a follow-up,
Yeah.
so I know we've talked about it before, but rather than estrogen and progesterone, glucocorticoids are metabolized very differently in the male and female placenta. And in your Zika virus infected animals, you see a change in aldosterone biosynthesis. It'd be very interesting to just look at the male and the female placenta and what steroids they're producing in response to Zika virus infection, because a lot of the processes that you're seeing being modified may actually be, being controlled by changes in glucocorticoid bioavailability or mineralocorticoid access to the receptors. So it might be a very simple thing that would actually allow you a mechanism to define why there is that sexual benefit.
Yeah, that's a good suggestion. Yes. So in our experiment, the Zika virus infected placenta or benzoin exposed placenta, we also need to fully evaluate what's the impact of this exposure to the placenta in terms of the histology or what's their functioning. So that's under investigation. Yeah. Thank you.
I was wondering, well, great talk, thank you. I was wondering if your data on Zika correlates with other flaviviruses such as dengue, or if that hasn't been done, mainly because, especially in America, dengue is predicted to become a major public health concern over the next decade or decades. That might be an area that could easily get a grant, and if it hasn't been done yet.
Yeah, I see. So it's always a challenge for me because I put Zika in the grant and then everybody's saying, oh, the pandemic is gone. What is, right? So I think for your question, so the dengue virus is very similar to Zika virus. So, but when I'm searching for the area that how viral infection affect the sex dimorphism of placenta and as well as the offspring health, there's no much data about that. And then the data is all like a descriptive, no mechanism involved. And then because although Zika virus has peaked in 2016, right now it's 7 years past. So when they follow up the baby, they found a lot of problems in the baby. They found in the child's blood, they found a very different population of the neutrophil and the inosinophil, a lot of differential phenotype in the offspring. Then it reminds me that, okay, so when the pandemic peaks at some point, you need time to follow up the children to see what is the problem in the children. As the same story as COVID. So we have been 4 years post-COVID, and they found in the male offspring, they have more problems in the brain, and they have more problems to the vaccination. So this is really, I think it's a really hot topic that we just follow the phenotypes, but we trace back to what is happening during pregnancy, and then we link everything together. But it's still under investigated area, so we definitely need to look into it. Yeah, thank you.
Thank you, Dr. Zhang. And I think it's an exciting area with the sexual dimorphism. It's been for many years, the field researchers have been investigating just fetuses without going into the sexual dimorphism. And it looks like it's a very important thing to assess this dimorphism. Our next speaker is our external speaker, Dr. Asgi Fazleabas, is a university distinguished professor at the Michigan State University. He's the chair of the research in the Department of Obstetrics and Gynecology and Reproductive Biology, and the director for the Center for Women's Health Research, and the co-director of the Reproductive and Developmental Science Program at the Michigan State University. Dr. Fazleabas received his BS, bachelor's from the California State University, Fresno, and doctorate from the Reproductive Physiology from the University of Illinois at Urbana-Champaign. Following postdoctoral training in reproductive biology, cell and molecular biology at the University of Florida, Gainesville, Dr. Fazleabas joined the Department of OB/GYN at the University of Illinois, Chicago, where he held the rank of professor and the director for women's health and reproduction until October 2009. Dr. Fazleabas, who is at the MSU, is a principal investigator of a number of research studies funded by the National Institutes of Health and has a huge number of publications. And thank you so much for being here with us today. I appreciate what you've brought to the next. Thank you.
Use this. If you can. Great. So thank you, Jane, and thank you, Gil, for the invitation. It's nice to come across the state. Don't do that very often, those of us on the west side of the state. So a little bit of a switch. You've been hearing a lot about the placenta and the end of pregnancy. I've always been interested at the beginning of pregnancy. So what I'd like to do is sort of take you through a role for a molecule that we've been working on for many, many years. But I've always been fascinated with what that little early embryo does in the uterus for that very short period of time. How they synchronize? How do they make sure that there is a communication that goes on? And most of the pregnancies that are lost, about 75% of pregnancies that are lost, are because of implantation failure. So really trying to understand the synchrony between the embryo and the endometrium is really critical for us to move forward, not only with normal pregnancies, but also with assisted reproductive technologies, because that's sort of been stuck at about the 30 to 40% range for many, many, many years. So obviously trying to study this window in the human situation is it's impossible. So several years ago, we actually developed a mechanism to try to do this in a non-human primate model. So what we wanted to see was what is the dyssynchrony between the increasing levels of progesterone and the embryonic signal, in this case, chorionic gonadotrophin, when the embryo is coming into the uterus, and how does it impact the endometrium preparation for pregnancy? And to do this, we developed a simulated pregnant model almost 20 years ago now. And what we do here is put in a small cannula into the oviduct of these baboons, and I use baboons as a model system. And we infuse chorionic gonadotrophin during that 4-day window during which the blastocyst is free floating in the uterus. And what we can do is using an all-Z mini pump, mimic approximately what a blastocyst would produce during a 24-hour period. And this is also the time at which the corpus luteum is most sensitive to chorionic gonadotropin responsiveness to make sure that there is progesterone production. So by doing this for 4 days, then we can take the uterus and the tissue at 10 days post-ovulation, which would be the approximate time at which the embryo begins to attach to the uterus. And when we first did these experiments, one of the most interesting things was how dramatically each unique cell type within the endometrium responded to the signal from chorionic gonadotropin. The liminal epithelium underwent this very unique syncytial formation called the epithelial plaque reaction. The underlying stromal cells became myofibroblastic, and the glandular epithelium obviously again became highly secretory. And these became really important to try to understand what these changes are and how it's going to impact the migration of the embryo into the between the luminal epithelium, and how the underlying stroma is transformed to become a decidual phenotype. So using this model, we did a whole number of studies, both morphological, biological, as well as molecular. And over the years, we were able to show that this decrease in tight junctions, which is a unique response to this epithelial plaque response, does help to facilitate trophoblast migration. And for the free-floating blastocysts, the synthesis of glycogen within these cells is obviously really important for their metabolism. The myofibroblast phenotype was important to inhibit apoptosis because that has to be stopped to allow these cells to transform during the establishment of pregnancy. And again, there was increased sensitivity, increased secretory activity of the glandular epithelium. But what really struck us was the increase in Notch signaling, both in the epithelial cells, particularly in the luminal epithelium, and the correlation between the change in myofibroblast expression of alpha-smooth muscle actin and Notch1 expression, which had been shown to be important for fibroblast differentiation in other different cell systems. So the question we then asked was really, how does Notch play a role in implantation? And that's what we've really been focused on for many years. And if you look at the expression of Notch signaling and Notch ligands, they're highly upregulated both in the embryo as well as the endometrium during the process of adhesion and invasion. So that early event that's so critical for the establishment of pregnancy is really associated with increase in Notch, along with obviously the other molecules that some of you are familiar with and some of you do work with. So the question then becomes, what is the crosstalk and what is the role of Notch signaling that's from the embryo, as well as the epithelial cells and the underlying stromal cells? And my lab really has focused on what's happening in the epithelial cells and the stromal cells, particularly in relationship to allowing these cells to differentiate. So based on some of the roles of Notch, what we've been really exploring over the years is developing some transgenic mouse models, both knocking out Notch ligands and Notch receptors, as well as overexpressing Notch signaling to understand what might be happening at the time of implantation. And then validating the signaling pathway in terms of its relevance to the human situation, and looking at some of the pathological consequences with regards to endometriosis, recurrent implantation failure, and unexplained pregnancy loss in terms of the role of Notch. So what I'd like to take you through today is really show you some of the transgenic models and what we've been able to learn using these different transgenic models, and show you data that we've been doing in the human, because obviously the translational concept of this is really critical if you're going to understand what happens during implantation. So at the very beginning, when we started looking at Notch and just did a very simple immunohistochemistry, we were able to show that Notch expression is seen in both, not only in the rodent, but also in the human that's been treated with chorionic gonadotrophin, along with the baboon that's been treated with chorionic gonadotrophin. And what we see is a switch from epithelial expression to stromal expression in response to the embryonic signal and in response to the presence of an embryo in the rodent. So this upregulation during implantation early pregnancy is important for mediating stromal cell survival. And the absence of Notch decreases cell proliferation. It inhibits cell cycle checkpoints and also increases apoptosis. But the first model that we developed was a progesterone receptor Cree Notch knockout. And this was knocking out Notch in both stromal and epithelial cells. And what we saw was that there was a decrease in decidualization, but outside a small decrease in the first litter, pretty much these animals had no implantation failure and they had normal pregnancies. So it pointed to an effect on the decidualization, but that did not really tell us whether that was enough to prevent these animals from getting pregnant. So this led us to the question to find out whether there was compensation by other Notch receptors. And I showed you in the summary slide, there are a number of different Notch receptors that are expressed both in the epithelium and the stroma. And so we wondered what would happen if we now knocked out all of Notch signaling with regards to pregnancy. And for those of you that are not familiar with Notch signaling, it's a relatively complex pathway where the transmembrane domain of Notch undergoes through two cleavage divisions, one through ADAM and then through gamma secretase, to result in an intracellular domain of Notch that's then translocated to the nucleus, where it binds a coactivator complex very similar to steroid hormone signaling, and selectively binds to CSL or RBPJ as far as the rodent molecule is concerned to initiate gene transcription. So we thought that if we knocked out RBPJ, whether we would see differences. And when we first mapped RBPJ expression in the mouse uterus, the pattern of expression around the site of implantation and subsequent pregnancy was very, very similar to what we see with Notch. So you see it very early at the site of implantation. And as decidualization progresses, as I'll show you that Notch is important, its decrease is important to initiate the differentiation process. You can see that Notch moves away to the secondary decidualization zone, and it's in the primary decidualization zone during early pregnancy. So it really paralleled the expression of Notch1 that we had seen. And to do that, to try to understand what it might be doing, we again went to our progesterone receptor Cree mouse model. And in this case, we knocked out RBPJ in the uterus of these animals. And in this case, what we see is there's a significant decrease in implantation. And these animals, unlike the Notch knockout, which has a lower litter in the first litter and then subsequently catches up, what we see is when we knock out all of the Notch signaling, there is subsequent infertility and there is progressive infertility. These animals become subfertile by the second litter, and by the third litter onwards, they become completely infertile. And one of the things that does happen with these animals, they also fail, that I don't have time to show you, they also fail to undergo remodeling postpartum. So that probably also is one of the aspects that contributes to this pregnancy loss that we see in the RBPJ model. But we want to see what happens during early implantation. And to do that, in collaboration with Ripla Arora at MSU, who does this beautiful three-dimensional imaging of the uterus, what we saw was when we looked at day 3.5, you can see that there's these gorgeous uterine folds that have to form within the endometrium to allow for implantation to take place. But when we knock out Notch signaling under progesterone receptor Cree, you can see how disorganized that endometrium is. And essentially those uterine folds that are so important for allowing for the crypt to form, for implantation to take place, are completely disorganized. And you can see what happens here is that in these animals, the embryo loses its appropriate orientation, and rather than orienting towards the mesometrial, anti-mesometrial pole, what it does, it goes parallel, and it can no longer form those crypts that are important for the embryo to migrate into, contributing to the infertility, or contributing to one of the components of the infertility associated with this model. But as I said, with Notch signaling, it's a very delicate balance. So at the initiation of pregnancy, both for epithelial cell proliferation and initiation differentiation, Notch has to be expressed. And this then contributes to the early proliferative response in these animals. However, for these cells to undergo complete differentiation, Notch actually has to be turned off. So for differentiation to take place, Notch has to be turned off. And this is not unique to the uterus, because essentially in lots of cancer studies, the inability of Notch to be turned off, and that's why you can use gamma secretase inhibitors or now small molecule inhibitors that target both mastermind and RBPJ, that this has to be turned off to allow for the cancer to be suppressed. However, in both breast cancer, pancreatic cancers, overexpression of Notch contributes to the pathology associated with this. So since the differentiation component was also very important, our next question was, what happens if we now overexpress Notch? What is the consequence of this with regards to the establishment of pregnancy? So in this case, what we did was, again, using the progesterone receptor Cree model, we overexpressed the intracellular domain of Notch in these animals. And you can see that both the GFP, which is a marker associated with this overexpression, and N1-ICD is highly overexpressed in these animals. And the response here in these animals is extremely profound. They're completely infertile, whether they're heterozygous or homozygous, and their uterus is very, very small. They essentially will have no pregnancies at all when you overexpress the intracellular domain of Notch under progesterone receptor Cree, which is that it's overexpressed in both the stromal and epithelial cells. And the progesterone receptor target genes are dramatically downregulated. And the reason for this, as I'll show you in a summary slide, is that the overexpression of Notch drives the upregulation of DMMT3 along with PU1, which allows for the methylation of the progesterone receptor. So essentially, overexpression of Notch also has an impact on the function of the progesterone receptor. And in contrast, if you look at the estrogen-regulated genes, the target genes that are important during implantation, they're all highly upregulated during that window of implantation. It's a day 3.5, which tells you that there is an altered response, which is so critical for the establishment of pregnancy. And the other thing that we saw here was also that these animals have no glandular development. And so again, the differentiation process, progesterone is turned on at about day 4 postnatally. That's so important for the development of the glands in these animals after parturition. But by overexpressing Notch, what we find is that the glands never develop, but instead the luminal epithelium maintains a glandular phenotype. So rather than expressing FOXA2, which is a marker of glandular development, they express FOXA2 in the luminal epithelium and not in the glandular epithelium. And you can see there's absolutely no glandular structures in there. And if you also look for LIF expression, the LIF is also maintained in the luminal epithelium and never expressed in the glandular epithelium. And if you look at the implantation process in these animals, here again, you can see how small that uterus is. And the blastocyst is always free floating in the uterus, and it doesn't implant and invade into the uterus like it should at day 5.5. So essentially, the overexpression of estrogen responsive genes, together with the absence of glands, has also a very dramatic effect in terms of allowing this free floating blastocyst to attach and invade into the uterus. And as a result, these animals are consistently infertile. It also has an impact on the vasculature. And here again, there's some 3-dimensional images done with retina. And you can see that in these animals, when you overexpress Notch, there's absolutely no glands. You can't see any of the glands here, which are labeled in red with keratin. But more importantly, if you look at the vasculature by staining with PCAM, that the vasculature is also markedly affected in these animals when you overexpress Notch. So a whole sequence of events associated with inappropriate development of glands, together with the inability of the stromal cells to differentiate, as well as the absence of vessels, all contribute to the infertile phenotype with abnormal overexpression of Notch1, the intracellular domain of Notch in these animals. So I'm summarizing a whole bunch of work here, but basically what we find is that by overexpressing Notch, which can happen in pathological situations, these animals have implantation failure, particularly because of the inability of these cells to undergo their differentiation, and they have decidualization failure and gland failure, both of which are so critical for the establishment of pregnancy. And the overexpression here in terms of suppressing progesterone responsiveness is because of the methylation of the progesterone receptor under the presence of high levels of Notch, which then suppresses progesterone signaling in the endometrium, both in the luminal and epithelial systems, and maintains this inordinate, inappropriate expression of estrogen-regulated genes during the implantation window. So, as I showed you again, that it's not just stromal cells, but it's also the epithelial cells. So the second question we then asked is, what happens if you now do the same experiment, but under lactoferrin Cree, which is epithelial specific? What happens if you now overexpress Notch, the intracellular domain of Notch, again, under lactoferrin Cree? And here again, if it's epithelial specific, whereas progesterone receptor is both epithelial and stromal, here it's epithelial specific. Again, what we see in a breeding trial is that these animals do not get pregnant. And in this case, what happens is that there is a huge effect in terms of the disorganization of that endometrium, and particularly the epithelial cells. And these animals, even if you give them lift, which you can rescue a lot of pregnancies in other model systems, lift does not have an effect in these animals, and they do not get pregnant. However, if you knock out the canonical response to Notch by crossing these animals with our RBPJ knockout, which means you take away the ability of overexpression of Notch to continue its signaling process, these animals are perfectly fertile, they get pregnant, and they don't have any problems. So it's a canonical response to the overexpression of Notch. And if you remove this, these animals are perfectly normal, they get pregnant, and they keep their litters with no problem at all. But on the other hand, if you now look at what happens to their endometrium again, you can see that there is also a disorganization, pointing to the fact that expression of Notch appropriately in both the epithelial cells and the stromal cells are really important for allowing that endometrium to develop in its appropriate manner. And here again, you can see that the folds are altered. And in this case, there is even a more complex reorganization, an abnormal reorganization of the endometrium because of the changes in the way the epithelial cells form. These animals have glands because lactoferrin Cree turns on at day 21. So there's no effect, no problem with the development of glands. But they also, you see this very convoluted effect of the luminal epithelium, particularly in these animals. And the morphological, three-dimensional morphological architecture of the endometrium is dramatically affected. And in a similar manner, if you look again to see what happens with the estrogen-regulated and progesterone-regulated genes, we see that unlike the overexpression under PR3, where all of the genes are downregulated, here we get a selective increase in MOC and LIF, and lactoferrin, sorry. and a selective downregulation of amphiregulin and Indian hedgehog, which are the early response genes that are important for signaling downstream for allowing decidualization to take place. So there is a selective downregulation under the lactoferrin pre-knockout overexpression as opposed to the PR3 overexpression. And in the RNA-seq analysis, again, just to summarize what we've done, the biological processes that we see are different in the luminal epithelium as opposed to glandular epithelium. We see abnormal cell proliferation and cell cycle genes that are upregulated in the luminal epithelium, whereas the glandular epithelium is hyperproliferative, and with an increase in estrogen sensitivity in these animals. So when you overexpress Notch now in the epithelial cells, where you normally see this beautiful differentiation of the luminal epithelial cells and the glands that are important for the establishment of pregnancy, the secretion of lymph, in the infertility phenotype that we see with the lactoferrin pre N1 ICD is essentially that the epithelium doesn't reorganize appropriately. The epithelial cells are hyper-responsive to estrogen. And as a result, you don't see this differentiation, and the ability of lymph to rescue this response is diminished. But also what's important is that if you take these epithelial cells and make organoids out of them, they're the best things you can do to create organoids. They proliferate like crazy, and they form these beautiful organoids that will allow you to really study epithelial function and epithelial structure. So from a model system to study epithelial cells in an organoid system, this is a really, really neat model. So it's not good for studying infertility, but it's great for studying glandular function. So now let me switch to decidualization. And essentially, obviously, this is an important response that's really important for promoting embryo implantation, for controlling trophoblast invasion, and fetal allograft acceptance. In rodents, the decidualization response requires the implanting embryo or a physical response, whereas human decidualization is a little bit more complex. You see a pre-decidualization response at the end of every menstrual cycle towards the latter phase of the secretory phase. And essentially, if that doesn't get accelerated by the presence of an embryo or an embryonic signal, essentially what you end up with is with menstruation. And we know from a number of studies is that impaired decidualization really contributes to significant preterm defects in women, along with other aspects that are consequential in later pregnancy because of failed decidualization. So essentially, this is one aspect of pregnancy that we can study in the human situation, because it is easy to get these stromal cells, it's easy to isolate them, it's easy to transform them, and we can then really study the consequence of decidualization with regards to disease processes. So for the second part of my talk, what I'd like to do is switch to what studies that we've done as far as using these stromal cells to try to understand its implications with regards to human pathology. But just here, summarizing again, that if we knock out the Notch pathway, no matter which Cree we use, we essentially end up with decidualization failure in these animals. So our question then is, what happens with regards to the human situation? Do we see the same mimic with regards to early response to allow these cells to transform, and a late response to prevent these cells from undergoing decidualization? So in this case, what we did was basically use human stromal cells, and we knocked down Notch expression prior to stimulating these cells with estrogen, progesterone, and dibutyrocyclic ANP, which is the classic cocktail that you use to transform these cells in vitro. So these cells are seeded, treated with an shRNA for Notch1, and then 5 days later, we start the experiment where we induce these cells to undergo decidualization. And you can see when we do that, the markers of decidualization, both IGF-BP1 and prolactin, are significantly downregulated when we knock down notch prior to the stimulation response, showing again that the early expression of notch is important for stromal cell differentiation. However, if you do the reverse experiment in this case, where we treat these cells with the mixture, allow them to transform essentially for 2 to 3 days, then we knock down notch, it has absolutely no effect with regards to decidualization, pointing to the importance of this myofibroblast differentiation and notch expression to allow these cells to transform into a decidual phenotype. And you can also look at this from a physiological standpoint, is that if you take these cells and treat them with either estrogen, progesterone, a combination of IL-1 or dibutyl cyclic ANP, you can see is as these cells transform into the decidual phenotype, it generally takes between 6 and 12 days, that notch is downregulated. Both notch 1 and notch 4, which is also transcriptionally regulated by notch 1, they're both downregulated as these cells transform into a decidual phenotype. And if you then look to see what happens with regards to a process that might be suppressing notch, you see that NUM, which is an inhibitor of notch signaling, is highly upregulated in these cells as they undergo decidualization and get about a 20-fold increase in NUM expression as these cells are transforming into the decidual phenotype. And we can also do the reverse experiment that we did in the mouse model, where you can now overexpress the intracellular domain of notch in these stromal fibroblasts and now induce them to undergo decidualization. And you can see that when we overexpress notch in these cells, just like when we suppress them in the early response, these cells do not transform and do not express IGF-BP1 and do not undergo decidualization. So in terms of what are the potential consequences of this with regards to various diseases? So my lab also studies endometriosis, and we've studied endometriosis a number of years. And disordered decidualization with regards to endometriosis is that you do see an accelerated senescence in these cells. You see impaired differentiation because these women generally do have decidualization failure, and progesterone resistance is a classical response that you see in the endometrium of these women with endometriosis. So we then ask the question, what happens with regards to decidualization in the context of endometriosis? And we go back to our baboon model, which can also induce endometriosis, and it's a physiological type of endometriosis that resembles peritoneal disease that we can create in these animals. You can see that both notch 1 and notch 4, together with the ligands and the transcriptional regulators, are all downregulated in response to endometriosis in these animals if you isolate the stromal fibroblasts. And this is very similar to what you would see in the human. And if you take stromal cells from women with and without endometriosis, you see the same pathways, the same genes that are downregulated. And by immunohistochemistry, you can show that notch 1, notch 4, and HES5, which is a downstream transcription factor induced by notch, are all downregulated during the mid-secretory phase, during the window of implantation in both women and baboons with endometriosis. So what then regulates this? So SIRT1 is known to be, it negatively regulates notch 1 expression. And it does this by de-escalating the intracellular domain of notch. So when we look at our baboon model and humans with endometriosis, you can see that in these, the stromal cells that we get from both women and baboons with endometriosis and do not undergo decidualization, and you look for the expression of SIRT1, you can see that in both women and progressively in baboons, and that's the advantage we have, is that we can induce disease in these animals and then test changes during the progression of this disease. You can see that in women with spontaneous disease or progression of endometriosis in the baboon model, you see an increase in SIRT1, which correlates with the downregulation of Notch expression in the stromal fibroblasts, and then thereby prevents these cells from undergoing the decidualization response. So normally what happens during decidualization is that the stromal fibroblasts are stimulated. So FOXO1 is an early transcription factor that's important for allowing these cells to transform. And generally, under normal physiological situations, these cells will transform into desferal phenotype, secrete prolactin, IGF-BP1, and interleukin-15, which then allows for the recruitment of UNK cells to clear out the senescent cells. So some elegant studies from Jan Brosen's group has shown that there is a ratio of senescent cells and normal destrual cells that are important for the establishment of pregnancy. Overexpression of senescent cells are associated with the recurrent pregnancy loss. And in this case, if everything is normal and everything goes along, you have the recruitment of the NK cells to clear out the senescent cells to allow these destrual cells to essentially continue and develop. So we wondered whether part of this is that decrease in Notch signaling, which again can inhibit FOXO1 from our earlier work, will prevent these cells from undergoing the destrual phenotype and then increase the number of senescent cells, which might also contribute to destrualization failure because the lack of IL-15 together with the absence of the upregulation of FOXO1 could contribute to this change in the ratio of both the destrual phenotype. So is this real? Does this ever happen? So we can go back and take a look at the expression of lamin B1, which is a marker of senescence. And you can see here both by quantitative RT-PCR and immunohistochemistry, you see a real marked downregulation of lamin B1 in the endometrium of women with endometriosis. And you can do the same experiment by just simply taking stromal fibroblasts, knocking down Notch 1, and you can see a consequence of that is the decrease in lamin B1 and an increase in P21, both markers of senescence. So the decrease in Notch could be one of the mechanisms by which this is contributing to the increase in senescent cells that you see in these women that are undergoing destrualization failure. So our current work is really looking at what regulates and what upregulates SIRT1. How does it then repress N1-ICD, and trying to understand the mechanisms of cellular senescence that might be associated with destrualization failure. So that's ongoing work in the lab right now in trying to look at these pathways and seeing how these pathways are affected and how does disease then affect these pathways to then contribute to destrualization failure. Then the other one that we've been working with, this is mostly with Carlo Ticconi and his group at the University of Rome, in terms of whether the destrualization response is also affected, does also contribute to recurrent implantation failure and recurrent pregnancy loss. Again, based on some of the work that his lab has done and others have done, where we know that there is a change in immune dysregulation, the inability of these cells to transform because of the increase in senescence and altered gene expression. So we wondered whether this might be a process through which there could be a contribution to these pathologies that you see in women with recurrent pregnancy loss and recurrent implantation failure. And when we go back and look at RBPJ, which was the gene I told you that was important for the initiation of destrualization, you can see that in control women, RBPJ is highly expressed, whereas in these women with recurrent pregnancy loss, it's completely reduced. And then what you see is if you look at the inflammatory component, in this case, interferon gamma, but if you look at all macrophage infiltration and other immune genes, they're all upregulated in these women with RPL. So what is it that's upstream of this that might be associated with Notch signaling And we looked at EGFL7, which is an activator of Notch signaling, early activator of Notch signaling in these patients with both recurrent implantation failure and unexplained pregnancy loss, because Carlo has a large clinic in where he gets these women, he works with them, and they come to him for treatment. And you can see both in proliferative and secretory phase, EGFL7 is highly downregulated in these women. And you can again go back and take stromal cells from these women and look to see what happens with regards to the downstream markers of destrualization. They all have failed destrualization because they have a decrease in EGFL7, which is an early activator of Notch. So you suppress early Notch signaling, and as a result, the destrualization response is dramatically altered. So here again, in these women, what we find is that there is an effect of destrualization. The normal process of where cyclic AMP is upregulated, normally through prostaglandins in the normal uterus, and EGFL7 and the intracellular domain of Notch working together to upregulate the genes that are important for this process and the transformation of these cells. But in the context of disease, what we find is that both the early activator together with Notch, they're all significantly diminished, and that in some way might contribute to the destrualization phenotype that we see in these women. A lot of work to do. This is just a paper that was just published a few months ago, and really getting into trying to understand the role of EGFL7 as well as the crosstalk between Notch is work that Carlo's group is doing in collaboration with us. So what I hope I've done is given you kind of a journey of how we've taken one molecule and try to understand using various model systems and bring it back to the human situation and see whether we can understand some of the complexities associated with the pathologies that we see in women that have pregnancy failure. So with that, obviously, I just do a lot of talking. These are the people that do the work. So the Notch work was originally begun by Yalda Afshar, probably one of the most brilliant graduate students, MD PhD students I ever had. She's now an assistant professor at UCLA. Mike Strug, another great dual degree candidate who's now a fellow at Stanford in reproductive endocrinology. And Renbi Su, who's now a professor at Guangzhou in China. They were really the early initiators of these studies. And then my current group, people that work primarily on the Notch signaling, Ariadna, Jenna, and Young, who's done all of the senescence work. And obviously, you know, science is a village, and it takes everybody to do all the work. So I've been very, very fortunate over the years that I've had great collaborators. And the whole story of Lucio Mille, whole story of Notch began with my friend Lucio Mille, who's the director of the Cancer Center down at Louisiana State University now, but we were both colleagues at the University of Illinois. And this whole story came about over a bottle of very, very nice Italian wine in his backyard. And because he was looking at fibroblast repair and wound healing. And we were talking about what we had seen with our early work in the baboon and the change of the myofibroblast. And he says, "Phew, that looks like wound healing. Let's look at Notch." 23 years later, we're still looking at it. So thanks again, and I'll be happy to answer any questions.
Let me ask you a question. Thank you for this amazing talk. So the picture that you had showed in the beginning about the uterine folds, is that what kind of imaging is that?
So it's actually laser confocal microscopy. And so what Ripla does, and she takes the whole uterus and essentially fixes in a DMSO-methanol fixative. And then you can use this to penetrate antibodies. So basically she can do whole mount analysis, and using these antibodies with the fluorescent probes, then they're imaged using laser confocal microscopy. And she first developed this in 2016. And that was the first paper in development, which has had 2 or 3 papers just recently. But it's a beautiful technique. It just really is, gives us so much more information than anything we can do in 2 dimensions. Yeah.
Yes, actually, like, there's a question that always comes up with PGR3 and the fact that, it's lovely that you have it in the uterus, but it's pretty much all over the reproductive system. Do you find that to be maybe a problem, or how would you answer that question?
So I mean, it's only one allele, right? So there's always progesterone responsiveness still available there. And the 2 tissues that seem to be most affected with the PR3 is the uterus, the mammary gland, and now some leakage in the ovary. But in general, other than that, you don't see a lot of pathologies in any of the other tissues.
Then the other question I had was, with the PGR3, you said the word actually leakage. There's with every Cree that I've worked with, you have some that are fantastic, every cell is exposed, and then there are others that over time. So I was wondering whether those 3 years, 3 different periods where the mice are being more and more susceptible to implantation failure could be a accumulation of more cells that are affected along the way with breed. There's of course the idea that over the long term the uterus is also getting less responsive. So
I hadn't really thought about it, but from looking at the work that Franco and John Leiden and then did over the years, it doesn't seem to be a dilution effect.
yeah.
There could be some leakage effect, but there doesn't seem to be any dilution effect. But the thought, I haven't really thought.
It'd be nice to look at it, test it, see, whether those trees. I know it's some of the trees that I.
So the only thing I can say is if you look by immunohistochemistry at the expression of whatever we're trying to knock out, it doesn't seem to change over time.
Okay,
So
no, that's.
if you're staying,
always good.
if you're staying at the beginning or you're staying at the end, they're about the same.
Yeah, one more question.
Now, my question is, as much signaling we looked at in other tissue.
Oh my
apart from
goodness, yes.
the endometrium.
Oh,
So the reason why I asked that question is that something that's always bothered me when we talk about implantation and the endometrium. Because you have pregnancies in other places. You have pregnancy in the tube, that no endometrium there. You have pregnancy in the peritoneum. You even have pregnancy after hysterectomy. And live birth have been born after pregnancy after hysterectomy. So my question is not signaling the answer to implantation rather than the endometrium, given that you have implantation in all these other tissue.
Oh no, I mean, implantation is so complex that there's no one, NIH wants you to focus, so that's focus. but no, I agree with you. It's not the only answer. It's just one molecule that's important for a number of processes that we think are critical for the establishment of pregnancy. But no, the complexity of the system is amazing.
Thank you. Let's bring our first external speaker for the day, Dr. Fazleabas from Michigan State University. And this is an amazing part of the symposium. We have the entire spectrum from conception, implantation, all the way till parturition, and we'll also have some neonatology and children aspect of the afternoon. And we're just going to take a short break here, and appreciate if you can be here a little before 11:15 when we will have our keynote speech for the day. Yeah, thank you.
That will be served over there. You can start.
Okay, so I would like to first acknowledge our platinum sponsors from Natera and Rapid FFN, and I just if you could take a minute or two, and perhaps if you want to say something to you.
Yeah, I'm this is Madeline. We're with Natera. Genetic testing is what we do. So we've had the #1 NIPT test on the market, which is Panorama. Horizon is our carrier screening, Empower, our hereditary cancer, Anora for miscarriages, and then Vistar is our single gene IVT test. And if anybody has any questions, our booth is out there. We can address anything further on any genetic testing. You guys do that.
Thanks,
Thank
guys.
you.
Yeah, thank you. Yeah, and hi everyone, I'm Jill McDougall. I'm with Hologic and the rapid FFN or FFN test. You know, good question earlier. Rapid FFN and FFN are the same. I've got some new data that y'all might be interested in. Number one, it's on the new data linking inhaled corticosteroids to potential neurological health issues later in life if mom receives them but goes on to deliver at term. And so, you know, that's been a big hot topic. Of course, that's a great rule out test for preterm labor. So just swing by and I can give you guys that info. And I'm always happy to do journal clubs and residency education and anything you guys need. So just let me know.
Thank you.
Yeah, thank you guys.
Our keynote speaker sponsors are these two companies, Natura and Rapid FFN. And I'd like to introduce our keynote speaker, Dr. Ahizechukwu is the Director of Research in the Division of Maternal-Fetal Medicine and an Associate Professor in the Department of Obstetrics and Gynecology at the Johns Hopkins University School of Medicine. Dr. Eke earned his medical degree from the University of Calabar School of Medicine, Nigeria, and completed 2 residencies in obstetrics and gynecology in Nigeria and in the Michigan State University. Dr. Eke continued his training at the Harvard University to earn a Master of Public Health in Health Policy and Management. Dr. Eke completed a dual fellowship in maternal-fetal medicine and clinical pharmacology at Johns Hopkins University School of Medicine and completed a Ph.D. in clinical investigation, clinical pharmacology at the Johns Hopkins University School of Public Health. Dr. Eke has served at the American College of Obstetricians and Gynecologists and the Society for Maternal-Fetal Medicine (SMFM) in many ways. Dr. Eke currently serves as the ACOG Clinical Guidelines Committee and on several other national and international OB/GYN committees. Dr. Eke has over 100 publications, cited peer-reviewed research publication, has won several teaching and research awards. And he was just showing me yesterday about his some of the work that he's doing with the White House and things that they have released in the past week, thanks to the amazing work of Dr. Eke. It's our pleasure to have such a renowned speaker with us today, and welcome back to Michigan.
Thank you, DA. I want to start by thanking the department for inviting me to give this talk. And I want to thank you, Dr. Moore, Dr. J. Ramadoss, and then also Alisa, who I've collaborated with quite a lot. I'm going to give a talk which is going to be a lot of deviation from what you heard this morning. I like basic science, I love basic science, but mine is purely clinical. And the goal is to kind of whet your appetite on the fact that we don't have great therapies for preventing preterm birth. We're still far away from the ideal. So that you guys in basic science, because that's where it starts, can begin to think of how to make things better so that we will get new therapies, because the ones we have right now, they don't work as well. I am a pharmacologist, so some of the things I will talk about would kind of be around drugs in pregnancy, especially vaginal progesterone, as well as some other progestogens that we've used to treat preterm birth. I'm excited to be here for 2 reasons. One, I trained here at Michigan State. I was here for 4 years, and I actually did some rotation at Wayne State with Ramadoss when he was still here. And then also because this state, and especially this institution, has contributed a lot to this topic. And you will see me cite a number of papers that came out of Wayne State in my talk. So I don't have any conflict of interest, but I was part of the what I call the gang of 16 that were responsible for taking out the drug called 17-hydroxyprogesterone carbapenem, which we popularly call Makena. It wasn't because we wanted to take it out by default, but it was just that based on the evidence that we had at the time, Makena was not appropriate. And so, as you probably know, it's no more part of this, of care for managing preterm birth. It's unfortunate because Makena is the only FDA-approved medication in pregnancy as a whole. And so right now we do not have any FDA-approved medication to manage pregnancy complications. Quickly, I'm going to just talk briefly about what we know about the epidemiology of preterm birth in the US, some of the factors that predispose to preterm birth, as well as some of the predictors we look at when we manage these patients. I'll define what a short cervix is because, as you know, it's one of the biggest predictors of preterm birth, how we evaluate a short cervix. And then I'm going to go into just some clinical scenarios so that you understand how we manage patients who have had prior preterm deliveries and see that there is no best approach to that there are still a lot of research opportunities right there. I'm going to review a couple of randomized trials and then a few meta-analyses that have been done. And then at the end of the day, I will conclude. So I must emphasize that preterm birth continues to be a significant cause of morbidity for the neonate and a critical public health problem in the US. Approximately 10% of babies are born preterm. As you can see from the graph right there, you see that the preterm birth rate in the US continues to go up. The latest figures show that it is still at about 10.5 to 10.9. And so you see the drop between 2020 and 2021. We think that was due to COVID because obviously people were not going to the hospital as they used to. And so it picked up again after COVID to go up to 10.5. So currently about 10.5 to 10.9 rate. This is critical because it accounts for about 20% of infant mortality in the US. And even more importantly, it leads to several long-term complications, as well as developmental issues in the babies. And so preventing preterm birth is something that we should do. However, we do not have any safe or effective medications to treat this condition. So treatment of a woman with preterm birth with progesterone is based on risk factors. Just like anything in medicine, there are risk factors that predispose women to this disease. And we know what these risk factors are. Generally, we classify them into maternal risk factors, which are factors in the mother, as well as what we call socio-economic risk factors, which are things that are related to lifestyle, education, income, marital status, as well as stress. What we do not know actually is the pathogenesis of preterm birth. What causes preterm birth? As you can see from here, the pathogenesis of preterm birth is very, very complicated. It is so complicated yet not well understood. Therefore, currently, we say preterm birth, we refer to it as a syndrome and not just one disease, because if you target one area, you're not doing justice. So we look at preterm birth as a general syndrome, and that way it helps us to manage it better based on what we know at the moment. So how do we predict treatment? How do we predict what to give and how to treat these patients? There are four major factors that determine the type of treatment with respect to pharmacology that we give these patients. The length of the cervix, if they've had a prior preterm birth, their parity, meaning is it their first pregnancy or is it their subsequent pregnancy? So the way a first-time pregnant mom behaves, it is different from the way, someone who has had 2, 3, 4, 5 pregnancies behave. And so that also has a direct implication to their risk of preterm birth, as well as if this is a single pregnancy versus a multiple pregnancy. So these four factors play huge roles in how we determine the treatment we give these patients to prevent subsequent preterm birth. History of prior preterm birth is by far the most significant risk factor for a subsequent preterm birth, meaning that if someone has had a preterm birth in her, in her lifetime, her risk goes up. And that risk goes up with respect to the number of preterm births she's had. So if she's had 1, it goes up by 20%. She's had 2, it goes up by 40%. And that risk keeps going up with subsequent risks of preterm birth. So it's good to understand that so that we can use that to see how we treat. A short cervix also is another risk factor. And this has been studied by several authors. So we see that it is a powerful preterm birth predictor, and numerous studies have looked at this from the US to several parts of the world, in Canada, in France, here in the US, and several other places. And we see that there is actually a meta-analysis of several studies that have shown objectively and very strongly that a short cervix is a predictor of preterm birth. Very, very close to what you would get with someone who has had a history of prior preterm birth. So that means that if someone has a prior history of preterm birth and then now has a short cervix, you can almost tell that patient that you are going to deliver early, and you will be right in almost 70 to 80% of the time. So we usually measure the cervical length during pregnancy. One of the things we do universally. So Wayne State actually was one of the centers, not one, was the first center in the US that started to do this across pregnancies, meaning that every woman who gets pregnant and comes to see us, somewhere between 16 and 24 weeks, we measure the length of the cervix, because it's been shown that that length of the cervix predicts her risk of preterm birth. Now, the optimal time to measure that length is somewhere between 16 and 24 weeks of gestation, because we've shown and we've seen that cervical length measurement in the first trimester of pregnancy, at a time when we measure what we call the nipple translucency, so that is the neck of the baby, to help predict the risk of syndromes like Down syndrome. So if you measure the cervix at that time between 11 to 14 weeks, it's less accurate. And so we usually do it between 16 to 24 weeks of gestation. Now, how do we do it? So the Society for Maternal-Fetal Medicine has established standards for measuring the cervical length. And this is important because if you want to do a research on cervical length examination or anatomy or anything like that, you must follow these steps on how to measure it properly. So you can tell, because once you say it's short, several things happen after that. And so basically, there are two ways we measure the cervix. You could do it from the abdomen. You could do it from the vaginal opening. The vaginal examination is way better than the abdominal exam. And when you want to get that exam in the vaginal route, we like the bladder to be empty. Because if the bladder is not empty, it can pull up the cervix and make it look longer. And so they empty their bladder, and then we follow these steps to try to get that measurement. We usually measure it 3 times. The best measurement is the one that is the shortest. That's the one you choose, because that's the one that would actually tell you if that risk is high or not. SMFM developed something called a CLEAR program, which is the cervical length exam and review. It's an online program. If you want to learn how to do this, you can just go there. One of the things they do is that you have to submit 5 images that you've done on your own, and then they will review that. And if you pass it, you'll be certified on how to do this. I did it when I was a resident, and I learned a lot from it because it kind of helped perfect how I measure my cervix at this point. It basically, what you see is that the cervical length is measured, the total length, and then we measure the length of the cervix. If there is an opening, you want to know what is between that opening and the external cervical os, as well as between the opening and the internal cervical os, because the actual cervical length is what is below the opening, which is the one that you see on the arrow. So how do we define a short cervix? A short cervix is basically a cervix that is between 27 and 43 millimeters of length. The average length of the cervix during the first and mid trimester is about 3.5 centimeters. And we also know, like I've said multiple times in this talk, that the shorter the length of the cervix, the higher the risk of preterm birth. But to put this into context, studies have also shown that the cervical length, if it's less than 2.5 centimeters, about 20% of these women will deliver before 37 weeks of gestation. And if it's less than 15 millimeters, half of them will deliver early. So like I told you, based on that cervical length and or a risk of prior preterm birth, you can almost always tell a patient that you are going to deliver early and you will be right. Now, treatment. So how do we manage patients who have had a history of prior preterm birth or short cervix? Well, there are 3 major ways we do that: medications, cyclage, which is a stitch around the cervix, and then the use of pessaries. I'm not going to talk about pessaries or stitch. I'm going to limit my talk to progesterones, and more specific to vaginal progesterone, because as you probably would see, we do not use the injectables anymore. Vaginal progesterone is a drug that we do not allow people to take orally because it has a very poor bioavailability. So if you take it, only about 20% of it gets absorbed and goes into the bloodstream, and that's not what you want. But and this is basically because of the high first pass effect in the liver. It has protein binding that's also high. So again, taking it orally will make only a very small fraction to get to the cervix where you want it. So that's why we give it vaginally. Now, it reaches its maximum plasma concentration in about 12 hours after you place it vaginally. And then by about 16 to 21 hours, it begins to leave the system, which is why it is dosed daily to be able to get that effect. It's eliminated by tissue metabolism. And so even with disease states, you don't have any issues with its metabolism. It comes in different strengths as well as different formulations. But the commonest ones we have right now in the US are these four I've mentioned. The RAI guys, they tend to use the endometrine to treat patients with who have had IVF to support pregnancy till about 10 to 12 weeks. But we in maternal-fetal medicine tend to use more of prometrium, which is 200 milligrams, or crenon, which is gel, to try to prevent preterm birth. I'm going to just talk briefly about this drug because that was the one I started a committee that we kind of we removed license from. But basically, it was used up until early this year to manage patients with a history of prior preterm birth. It was an injectable drug, highly protein bound, highly lipid bound, had a very high volume of distribution, and also stayed in the plasma for a long time. But you would think that because of that, it would have a good effect. But again, we found that it was not effective. And so I'm going to go through what I call scenarios, looking at all the risk factors I talked about. How do we use these risk factors to triage patients, and how do we treat them based on that? So the first scenario, remember I said every patient who comes to this country, you're pregnant and you get to about 20 weeks, we measure your cervical length. Now we use that to triage the patient. So let's say we measure it, it's normal or unknown. It's difficult to measure. Sometimes it's difficult to measure. Unknown or normal length. We now ask ourselves, is it a single pregnancy or multiple pregnancy? So unknown cervical length or normal length, single pregnancy, and a history of prior preterm birth. So this patient, we've triaged this patient in this way. What is the treatment? Well, before April of this year, we used to give them 17 hydrogen progesterone caprate, and that data came from two big studies. The MISS trial, which was published in the NEJM a couple of years ago, which was the data that led to this drug being approved by the FDA. And then most recently, about 3 years ago, which is the prolonged trial was published. The two studies did not agree. Why the MISS trial said this drug was very effective, the prolonged trial said it was not, which was why the FDA convened 16 people to decide whether this drug was good or not for pregnancy. And so when we voted, this was 4 years ago, 9 of us said we should still keep it in the market, but we should find other alternatives. 7 people said we should take the drug out of the market. The company had a role in this. They had to either say they want to put the drug on the market or they want a second hearing. They decided that they wanted a second hearing because they felt that the first hearing was not appropriate. So the FDA agreed and set up a second hearing last year, October last year. This time, it was worse this time. 16 people said, pull the drug. Nobody said the drug should remain in the market. And so around April of this year, the FDA made an announcement, this drug should be pulled. But something interesting happened. Now, when the FDA said they are pulling their license for this drug, ACOG, which is the American College of OBGYN, and SMFM, Society for Maternal-Fetal Medicine, they're the two big bodies in the US that make recommendations for pregnancy on how we should manage patients who are pregnant. So ACOG came out and said, well, we agree that this drug is not effective, it should not be recommended. However, we think that vaginal progesterone can be used, should not be used because it has not been proven to be effective in this population of patients. Remember, normal cervical length, single pregnancy, prior preterm birth. We have not shown it to be effective in this population, therefore we should not use it to manage these patients who now have lost this drug because of not being effective. SMFM, on the other hand, said, yeah, we agree as well with the FDA, but we think that this drug is not invasive. It's a vaginal drug. It's something there. It may work, it may not work. And therefore, we should consider using it in these patients who have had a history of prior preterm birth and normal cervical length. And so the OBGYN community, there was some confusion. And so it led to we now going back to review the data to see what the data had. So are there trials that address this particular issue? Vaginal progesterone in this population of patients, one pregnancy, history of prior preterm birth, normal cervical length. Now, there's been a couple of them, and I'm going to just quickly review them. So this was a study that was published by Eduardo Fonseca from Brazil. They looked at vaginal progesterone in a group of patients. Some of them had short cervical length, some had normal. In the subgroup of patients that had a normal cervical length, vaginal progesterone was not shown to work. It was not shown to reduce the risk of preterm birth. O'Brien from, he's in Kentucky, he's at MFM at Kentucky. He did one of the largest studies ever, the use of this in vaginal progesterone in patients who had normal cervical length. Again, he showed that it did not work. But we had other smaller studies. This one from Pratima Maggi from India, 100 patients, 50 to drug, 50 to placebo, showed that if you use vaginal progesterone in patients who have single pregnancies as well as history of prior preterm birth, that it worked. Again, this is, if you can read Arabic, you understand that. But this was an Iran study that showed that if you do 100 milligrams of vaginal progesterone between 24 and 34 weeks, that it reduced the rate of preterm birth in this population of patients. Remember, normal length, prior preterm birth. And then we had Elsin, Sentigos in Turkey showing that it worked in their population in Turkey. We had Modi as well in India showing that it worked. And then we had Azam, who is a very good friend of mine, in Iran, who also showed that in his study that it worked. If you looked at the smaller studies, it worked in the smaller studies, but it did not work in the bigger studies. And so two other studies, the one by Norman, which I was fortunate to have attended the SMFM conference in 2016 when this paper was published, and we were shocked to hear that vaginal progesterone was not effective. In fact, someone asked Jane Norman at SMFM when she presented this paper, if her daughter was pregnant, she was going to give her daughter vaginal progesterone, and she said no, that vaginal progesterone doesn't just not work, that she felt that this drug was actually toxic to the babies. And this was one of the largest trials in UK as well as Sweden with respect to the use of vaginal progesterone during pregnancy. Caroline Crowder as well, in New Zealand, Australia, as well as Canada, subsequently did her own study and found out that it did not work in their population as well. And then the group in Egypt, Ahmed Mahmoud Abdul, showed that it worked. So it's surprising that the smaller studies showed that vaginal progesterone worked. The bigger studies showed that it did not work. And you know what next would happen, right? So when you have randomized trials, some studies you show effect, others you don't show effect. The next thing you would see in a few months would be something like this, a meta-analysis, right? So you want to pull all the studies together and see, okay, is this because of small sample size? Is it because of patient heterogeneity? What is happening? Why is it that we have some group working, others do not? So Augustine Conde is somebody who I revere a lot because he has published quite a ton of this kind of studies in pregnancy. And he worked with Romero when he was still here in Detroit. And they published this paper asking the question, if we do vaginal progesterone in this population of patients with normal cervical length and a history of prior preterm birth, does it work? So this is a forest plot, bringing all those 10 studies I talked about. And you can see that the point estimate on the right side was not significant. And so for the primary outcome of less than 37 weeks of gestation to prevent pregnancy or preterm birth, it did not work when we pulled all the studies together. For the secondary outcome of preterm birth at less than 34 weeks of gestation, also this drug did not show to work in patients with a history of prior preterm birth. And so you might ask the question, so maybe if we add other therapies to this, maybe if we do a cerclage or we do, a pessary, maybe it would have some kind of, effect to help to prevent these patients. We also looked at all the studies that had been published looking at progesterone, specifically caproate as well as vaginal, and showed that even when you combine those studies, there was no benefit in combining vaginal progesterone with cerclage in this particular population. We looked at it in two groups. So the first group was patients who had a history cerclage. The second group was those who had a cerclage for an ultrasound indication. And we also showed that even with this population, the risk or the benefit of doing a combined approach was not there, because there were no differences between patients who were just having just the drug versus those who had the drug as well as cerclage. And so, coming back to my question, someone with a history of cervical length that is unknown or normal, singleton gestation, prior preterm birth. Before now, it used to be the injectable forms. In 2023, November, we do not have any evidence to show that anything works. Can you keep your question till after I finish?
Oh.
Yes.
Yeah.
We do not have any evidence that anything works to prevent preterm birth with respect to progesterone in this population. So how do I counsel my patients who come, given that this drug was removed from the market? So basically, I reviewed the literature. I talked about, preterm birth, what it does to the baby, long-term, short-term. I discussed the trials I talked about, specifically those of them that looked at the injectable forms, the design, the population, the results, the conclusion. And then if they have time, and believe me, patients have done this, I refer them to the YouTube videos of the hearings that we were on. The first one was 8 hours, second one was 16 hours. And they go there and they listen. And when they listen, they understand why this drug was pulled, because some people felt it was political. But if you follow that, if you listen to those hearings, you will see why we did not feel that McKenna should be in the market. Now, after they review all this data, then we can now decide on their plan. So there are 3 things or more that you could do. So first is do nothing. We don't like that plan. There must be something to do, obviously. The second is you check their cervical length. So every week you bring them back, do a vaginal exam, look at the cervical length. And then when it's short, treat them with McKenna. That seems to be something that is reasonable. We put a stitch around the cervix. That is what most MFMs do in 2023, November. Another option is cervical length. And then when it's short, you give them McKenna. Obviously, based on all I've discussed right now, you would know that that is not what we do. So basically, we call it a shared decision making. You discuss with the patient that right now we don't have any good therapy medically that will work for you if your cervix is normal and you've had a history of prior preterm birth. We can do ultrasounds for you, follow your cervix, and if it becomes short, then there is a therapy for short cervix, which brings me to my second scenario, which is now your cervix is short, right? So the first scenario was a normal cervical length, prior preterm birth, and single pregnancy. This time, cervix is now short, okay? Still singleton pregnancy, but this time a first-time mom, right? So this is when vaginal progesterone has been shown to work. And the data is quite compelling. You know, it's been shown to work in this specific condition where short cervix, first-time mom, or first-time pregnancy, and singleton gestation. And that data comes from 3 major sources. So you have the one I discussed this trial before. The major people in this trial were actually patients who had a short cervix. Only about 10% of them did not have a short cervix. So in this Fonseca trial, which I'll review in a few minutes, you will see that vaginal progesterone was shown to work in this population. And then here at Wednesday, Sonia Hassan, who is now, I think, the wife of the mayor of Detroit, did this. She was the one that pioneered the Hassan trial. Is what we actually call it our field. And you also see that this study was also positive. And then, as you would expect, Romero and Conde did a meta-analysis to show that when you put this data together, that vaginal progesterone in patients with a short cervix was beneficial if they had a singleton pregnancy. And so just quickly going through the detail of the Fonseca trial, published in NEJM in 2007, they recruited from 4 countries, from the UK, from Chile, from Brazil, and from Greece. These patients were singleton. So they also included twins as well, but the twins were kind of very, they were just about, I think 20 patients who were twins in that study. They randomized these patients 1:1 between 20 and 24 weeks of gestation, gave them 200 milligrams of vaginal progesterone every night. Primary outcome, preterm less than 34 weeks of gestation. Secondary outcome, they looked at a bunch of things, which is what we do these days. So we lump all the issues in the babies as secondary outcomes, which is one of the issues with the FDA, why they did not approve McKenna, because they felt that these things should actually be primary outcomes. And gestational age should not be a proxy for these outcomes. And this trial showed that the primary outcome was very impressive. They showed that vaginal progesterone, when you use it, there was a 44% reduction in the risk of preterm birth below 34 weeks of gestation. 44% reduction in the risk of preterm birth below 34 weeks of gestation. They also, however, did not show any reduction in the risk of discovery outcomes. So all the outcomes in the babies did not show any direction towards being significant. And so this was a problem. So when this trial was published in 2007, people were worried. Say, okay, you had this good outcome for primary outcomes, but all the outcomes in the babies were not significant. They felt that it was because of power. So that now led Sonia Hassan in her own trial to now power herself for secondary outcomes. And so in 2011, this study was kicked off at the PRB here in Detroit, and they recruited patients across the US in 44 different hospitals across this country. Again, very similar to what the people in Brazil did. They randomized patients to 90 milligrams of progesterone this time, not 290 milligrams. And primary outcomes, again, quite similar, less than 33 weeks of gestation. Secondary outcomes, exactly what they did in the Brazilian trial. And they showed very similar outcomes. So the Brazilian trial showed a 44% reduction. Sonia Hassan showed a 45% reduction. So very similar outcomes, which is huge in obstetrics, right? Something that will reduce the risk of preterm birth by more than half, but by about half. We will like it. But they also now showed for the first time that the preterm birth reduction at 28 weeks was also quite significant. As you can see there, almost 50% of babies did not deliver preterm at 28 weeks. And also, they showed that the rate of RDS as well as ICU admissions were reduced drastically. And so that led for the first time for us in obstetrics to now say vaginal progesterone in this population led to a reduction in the risk of preterm birth by significantly, and also helped the neonates. And so that was why right now in obstetrics, in MFM, vaginal progesterone is strictly used for patients with a singleton gestation, no prior history of preterm birth, with a short cervix. So Romero and colleagues again did a meta-analysis to look at prior studies, and you can see that the study by Sonia as well as Fonseca made it up there, and then a few other studies as well. And they showed two major things. First of all, they showed that vaginal progesterone was effective in this population. But they also showed that if you use vaginal progesterone anywhere between 90 milligrams through 200 milligrams daily, that you achieve the same effect. So even though some trials use 90 milligrams, some use 100, some use 150, others use 200, the effect of the progesterone itself was not based on dose. That once they're able to receive some progesterone, that risk of preterm reduces by almost a half. And so the other question, which we now started asking ourselves was, okay, sometimes this service becomes so short that would you allow a woman whose service is getting short, she's on vaginal progesterone, it's not working, it gets short, opens, and then she delivers. We see this all the time. So even though vaginal progesterone is effective to prevent preterm birth in a primary gravid patient, can we use a seclage as an adjuvant? Now, some people looked at this as well. This is a study that was by Dr. Nakone at UIC in Chicago. He was a fellow at the time. And he looked at their patients and said, if you had a patient who is a primary gravid patient on vaginal progesterone, and you're checking the cervix and it's getting short and short and short, and she's on treatment, do we put a seclage? And he showed something very interesting. So they looked at their population. You see that patients who had seclage plus progesterone, they delivered at 34 weeks compared to those who did not at 27. So that was quite significant. And so they also went further to now show that even in that population, as you can see, the outcomes in the babies were also very significant. So everything they showed from primary and secondary outcomes were very significant in their population. The study by Suka was the second paper that came out just about the same time from Greece. They looked at the same patients who were followed for vaginal examinations with vaginal progesterone and cervical length, and were on vaginal progesterone for treatment, and now had a seclage. And they saw that they demonstrated that in women with a cervical length of less than 1 centimeter, there was benefit in using seclage. And that seclage prolonged delivery by more than 32 weeks of gestation, and also more than 34 weeks of gestation. And so this was now began to build data to say we might actually have some benefit to using a seclage in this group of patients. And so Begela, who is at in Pennsylvania, he's at the Temple University. I can't remember anymore. He now did a meta-analysis with his groups, in Pennsylvania, and showed that if you pull these studies together, that patients who had cervical lengths of less than 1 centimeter benefited the most from the seclage. So remember that these patients had a short cervix, no prior pregnancies, followed with ultrasound examination of their cervix. And when it gets to less than 1 centimeter, that's when he showed that they were actually beneficial. So the conclusion is that if you have a patient who has no history of prior preterm birth, has a short cervix, vaginal progesterone is the treatment for it. And this has been shown in the studies I showed you. The dosing does not matter. They should just get something, vaginal progesterone, it works. If their cervix becomes less than 1 centimeter, they will benefit from a seclage. That's the summary of the patients who have a short cervix and on vaginal progesterone. 17-OHPC does not work in this population, and it's been shown over and over again. This study also shows that seclage, when it's above 1 centimeter, does not work. So the only time a seclage will work in a woman with a short cervix is if it's less than 1 centimeter, because this study looked at those who were between 10 and 25 millimeters length. Third scenario, short cervix, singleton pregnancy. This time it's a multipara. So she's had prior babies, right? But no prior preterm birth. So what do we do in this situation? Well, it's very similar to what we do in patients with a singleton gestation. We don't have great data, but we looked at it and said, that is based on what we have currently. That is what is most significant to do. And while we don't have any RCTs that specifically answer this question, these two studies that are observational that I discussed previously, and this RCT and meta-analysis show that the same things would apply to patients who have a short cervix and are multiporous without any history of prior preterm births. The fourth scenario, so again, short cervix, singleton pregnancy, multiple gestation, but this time, history of prior preterm birth. So this is a group where things are just wishy-washy, right? So there's vaginal progesterone, there's seclage, there's both. Do we do one for the other? And there are no great studies, even though, like I discussed earlier, some of these patients were included in these 4 trials I had talked about earlier. Some of the patients in this group were in that trial. But there's no study that has looked at them as a solo group, looking at patients who had a history of prior preterm birth and multiporous. And so when we looked at data from these 4 studies, or these 3 studies and this analysis, you see that from the O'Brien trial, the Progress trial, Optimum, as well as the meta-analysis by Romero and colleagues, O'Brien showed that when you look at the primary outcome of composite of fetal death below 34 weeks, death of the neonate, as well as composite score at 2 years, he showed that there were no significant, if you look at them as a composite, even though the death of the neonate was significant, but as a composite, it was not significant if you looked at just them as a primary outcome. And so the outcomes in the secondary data as well were not significant as well. So what he's trying to show is that vaginal progesterone, when used in this scenario, was not significant. The study by Crowder and colleagues as well in Canada and Australia and New Zealand also showed that when the cervix is short and patients had vaginal progesterone versus placebo and were multiporous, they looked at the primary outcome as well, with the highest incidence being RDS. They showed that there was no significant effect of vaginal progesterone in this population, as well as on all the secondary outcomes. So vaginal progesterone did not work in this population. Again, Jane Norman, I talked about this. They did a similar thing, looked at their own population, primary outcome as well. They found that vaginal progesterone did not reduce any primary outcome, and also did not reduce any secondary outcomes as well. And so again, you will see this all the time, right? Once there are 2, 3 RCTs, somebody must do this, right? And so Romero now showed that when he now put all the studies, interestingly, he found that vaginal progesterone worked. So how is that? Like, you would think that if all the studies showed no effect, the pool study would not show an effect. But he did what we call an individual patient meta-analysis, which looked at individual patients going into this pool. And he showed that if the cervical length is less than 2.5 centimeters, that these patients will benefit from vaginal progesterone. And so right now, that's how we, so any patient who has a history of prior preterm birth and is multiporous, we would recommend and give them vaginal progesterone. And so this is my last scenario. Scenario 5, these patients, again, cervical length is normal. They have multiple gestation, prior preterm birth. One thing you will learn quickly is that once there's twins or higher order multiples, nothing works, right? It's unfortunate. This is, an RCT they did. Again, it did not work. If you give it intramuscularly, they looked at the meta-analysis. Again, nothing worked, right? Twins. Whether you give it vaginally or intramuscularly, it was not shown to work. And so, multiple gestation, no prior preterm birth. Again, once it's twins, whether you give it intramuscularly, it will not work. Short cervix, it doesn't work, whether you do vaginal or intramuscularly. And so I will summarize by just going through the algorithm again. So again, normal cervical length, singleton gestation, prior preterm birth. we don't have evidence for the use of progesterone. Normal cervical length, multiple gestation, prior preterm birth, nothing will work, right? Short cervix, single gestation, primary gravid patients, this is where we have a weak, right? Because vaginal progesterone works, and then if the cervix is less than 1 centimeter, a cerclage would work. Single gestation, multiple pregnancies, no prior preterm birth. Again, vaginal progesterone in proxy with cerclage if you think the patient will benefit from it. And then short cervix, single gestation, multiple, and no prior preterm birth. We've shown that vaginal progesterone, cerclage, or both might be indicated. And finally, normal cervical length, multiple gestation, prior preterm birth, nothing works. And so with twins, either you look at prior preterm or no prior preterm birth, nothing works. So in summary, I would just say that while progesterone has come to stay, we give it like candy, there is no good evidence that shows that it works, except in populations that first-time moms, short cervix. That is the only time current evidence shows that vaginal progesterone works. And so that opens up a lot of research avenues, because what I'm just telling you right now is that we don't have anything that works to prevent preterm birth. Thank you, and I'm happy to answer any questions.
Can we add another scenario that you cannot, how they handle the compound? Maybe the compound is light sensitive or air sensitive, or how they store it or something like that. Because you are looking on the output of the patient.
Right. So your
Maybe
question.
the problem is how you start the treatment.
Okay. So your question is about the quality of the progesterone that was used. Is this for the?
It could be light sensitive.
Light sensitive.
Some people just put it in the light before they use it, or air sensitive or something like that.
Right. So the company that was licensed to distribute this drug in the US in 2011 had a very good way to start. So I don't think that was the issue, because we also looked at that. We looked at whether they stored it properly and whether it was an issue with storage. And we quickly realized that it wasn't the issue with storage. And I hear you, it's possible that if you don't store it properly, it could affect its efficacy. But the big RCTs that I showed you, most of them, integrity of the drug was reassured. And so that it didn't work in those studies was not because of the drug itself, but because.
Maybe it's because nobody studied the stability or the air light sensitive. We have a lot of examples in the drugs, light sensitive or something like that, and they find it later.
So we at the back end, when we look at the data, we saw the data for this drug. And there are several things that we saw that I can't disclose, but the data for progesterone itself, injectables, was not the issue with light, because they tested the drug itself for people who received it and for the storage conditions. So people that were given this drug in hospitals that used it, we pulled samples from places and tested, and it showed that it was still active. The active component was still the same prior to giving it to the patients. So we don't think it was due to that issue. Again, it can occur, but at least for the studies I showed you, it wasn't an issue with light sensitivity or not.
Okay, why this drug? Why the focus of this drug? Is this the only one?
That's what I'm talking about. This is where we're stuck right now. So if you have better options, please study it. Progesterone is the only thing right now with respect to drugs that we use to prevent preterm birth. And we moved away from doing it intramuscularly because of what I showed you. It did not work. People are saying do it vaginally, but the data does not support it. It only supports it in one group of patients.
The FDA removed now from everything.
Yes.
Yeah. So they use that specific various.
Yes.
It's not under the specifications of the FDA.
So the use of vaginal progesterone is off label.
It's off level.
So if you're using
Also, the price went down. Of the-
oh yeah, absolutely, absolutely. All right, I'm gonna answer you. Yes.
So you mentioned that there were many doses used and there wasn't like an effect with the dose, and that the studies found it was either effective or it wasn't. Were any of the studies stopped early because it was detrimental?
Good. So very good question. So the one of the things that I forgot to say, and again I can take 10 hours to to say this. They showed consistently that in the short term, progesterone itself did not harm the mom or baby. Again, we don't know what might happen in the long term. So our removing approval for the drug was not because of safety. Safety was not the issue. There were a few things here and there that happened, but if you look at what happened, it wasn't different from what happened, with other drugs. The main reason why we put the approval was because of efficacy. It did not work.
So
That was why.
follow-up question to that, if it's not detrimental, then why not just give it? Because the effects of being born preterm are gonna be, I mean, like you said, we don't know the long-term effects, but we know the long-term effects of preterm birth.
Good. So that is why we have opinions that are dissenting between ACOG and SMFM. ACOG says, don't use it. We haven't studied it yet in this population. We don't know if it works. SMFM said, oh, we think it's safe. Use it, even though we don't know if it works, right? So you have a point, and that is why what I do is I throw it exactly like that in the patient. I said, hey, we don't know if it works or not in your situation, but we also have not shown that it is risky in the short term. If you want to use it, you can use it. And so patients decide, right? Yes, you.
Well, because that was my initial question. So
Sure.
you had mentioned one study that, with the 90 milligram, the lower dose, that they saw that with the preterm births, there were fewer babies that were admitted to the NICU. Do you think that that's just by chance variation results between some of the studies, or do you think that with that lower dose that really will be something that's beneficial?
So what I can tell you for sure is if you go to clinicaltrials.gov, there are lots of studies now looking at all these combinations I talked about to try to see if truly we're seeing an effect or what we're seeing was not stuff. But what I can tell you is that the studies were so heterogeneous. Patients were lumped with many risk factors and all that. So one of the things that we're trying to do now is to do clean studies, clean RCTs to show that either this worked or not. So it's possible that it will work, but for right now, based on what we know, we cannot say that 90 milligrams works better than 100 versus 200. Yes.
So, from my own point of view, guys, I believe there's a big difference between preterm labor and how the baby is doing. So my question is, has there been studies that looked at the babies to see whether in fact, yes, the preterm labor, the went into preterm labor, some babies do better than the other, depending on whether they have the drug or not?
Good. So you bring up a very critical point. And I must say this so that because we have a lot of researchers in this room, and I see how exciting we're looking at research and all that. One of the things the FDA is going to be looking at in the future is if you're doing a study on preterm birth, please move away from using gestational age as your outcome. The FDA is going to ding it at some point when you take your NDA to the FDA to start a study in humans. Just because they realized recently that we should not be using gestational age as a proxy for how this baby is going to do, right? So we should be using what the babies suffer, how they feel, function, and survive. And that was the change in the second study, the prolonged trial. The FDA refused for them to use gestational age as their primary outcome. They said use fetal outcomes. If a woman goes into preterm labor and delivers, use how that kid is doing as your primary outcome for us to know that if this drug worked, the kid will do better versus if it doesn't work. And that's the clearest example to tell us that your drug worked or not. And so that study did not work. And the FDA said, well, you now see why we put your drug, right? So future studies, the FDA is saying that people should be using fetal outcomes
those things you see, how the babies feel, function, and survive, rather than just using less than 34 weeks, 33 weeks, or 37 weeks as a proxy, that it's not shown to be the best thing to do. Yes.
I have a mechanical question. I
Yes.
was wondering if a woman who has delivered prior or a woman who has multiple pregnancies, does her cervix shorten at a faster rate? And then women who have never delivered, they just, or their cervix isn't shredded, for example, so it shortens slower. So is that why progesterone is more effective? That the cervix is just not as pliable in a woman who has never delivered?
Right. So very important question. So for women with twins, I can tell you for sure that their cervixes shorten faster than those who don't have twins. And it makes sense because obviously 2 or more babies it stretches out and you know that. But if someone has had a history of preterm birth versus someone who has not, the cervix of that person with a history of preterm birth, obviously, because one thing we know is that it starts with a short cervix, right? So most likely that cervix will shorten faster than someone who has not. And so the role of progesterone, which we don't even know how it works, that's also another issue, right? Because at the FDA hearing, the question was asked, so how does this drug work? Nobody knows. I can assure you, if you know how it works, tell me. Nobody knows how it works. So how can you say it works?
So
Exactly.
What about, has anybody looked at women who have not had a prior preterm birth, who now have a short cervix, and broken them up into women who
Nobody.
have an accelerated shortening versus, and that might be actually the reason why you're getting a 55% in some, or in different ethnicities or different populations.
Absolutely. There's a lot of research gaps here, you know, to be done. And I can tell you anything preterm birth would get funding from NIH because this is the commonest cause of death. It's true. This is the commonest cause of death in babies in this country. And so much of dimes will give you money, will give you money, NICH will give you money. We are desperate for something to work. We don't have a solution to treat this condition. Yes.
I have another question to you actually. Has anybody looked at services of women, maybe when they had hysterectomy or whatever, for that previous preterm labor to actually see the architecture?
Yes.
So what was?
Yeah. So actually, Molly Stout at University of Michigan, she just looked at that, and she's beginning to see that it's actually not an issue of. She's showing that it's more about the skeleton of the cervix, that there are broken fibers that happen, and so that's why they deliver late. And so that the question is, how does that translate now to treatment? So she presented last year at SMFM, no, actually this year at SMFM. She will publish it soon. But she showed that when you take those specimens from hysterectomy in women who had a short cervix, their cervixes were different structurally from those who didn't have a short cervix during early years of pregnancy. And so we're beginning to think that it's more of an issue of the fibers not aligning properly versus issue of function of progesterone. So if that is the issue, then progesterone should not be working, which makes sense, right? Yes.
So the current definition of short is 25 millimeters. We have shown that even in women that deliver a term, cervical length changes with maternal characteristics and gestation length. And we have shown that if you consider those maternal factors, including, parity and so on, what is short is going to be different for different women. And we have shown that the personalized percentile of shortness is a much better predictor, substantially better than actually raw cervical length. Do you foresee any future where we could do a clinical trial where the definition is not going to be 25, it's going to be, can be 30, it can be 20, depending, for that particular woman?
I agree with you 100%. The problem with that kind of studies is going to be how would you convince the NIH to do an RCT with everyone having their own?
How about saying that the personalized cervical length percentile, it's a much better predictor of birth, and I'll show you that today on different cohorts.
That would be exciting. I'll be happy to see that.
Can there be another clinical trial providing progesterone given that, given the history? I mean,
oh yeah, absolutely. Vaginal progesterone, like I said, it's not invasive. We don't even know if it works. So if you want to do an RCT right now, you can. So that is a good research gap right there. Use percentile scoring as your short cervix. Randomize people to 2 groups and see. Absolutely, it's something that you can do.
You still have time. Yeah. So the architectural pattern that's noted from hysterectomy specimen, is it possible for that architectural pattern to be determined by ultrasound? Has anybody looked at it? That's my first question. The second question is that if it is possible, okay, especially in patients with previous preterm bad, do your cycle. Progesterone doesn't work. Do your cyclic galley. Do it at 10 weeks. Why not?
Right. So good question. Unfortunately, the ultrasounds we have now do not. What they used at the University of Michigan to do their study was something else. MRI, yes. They use MRI to show that.
Yeah.
So ultrasounds will not show that. So that's what I'm saying. In the next couple of years, we'll get there where we'll see that structure.
Sure.
And if the structure is weak based on that MRI or some kind of imaging, then we'll do a cyclage. Yeah, absolutely. 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 Yeah.
Right. So, very nice presentation. I'm just wondering the variation that in this study, have they ever just considered the sex dimorphism of the like the male baby or female baby? Is that because in this one study, if they pull together all the data together with the male baby, female baby, then we may not see a significant impact because some are working and some are not working. So did they ever?
They've not. Trust me. I know this literature very well. They've not. So again, it's a research gap.
That we should.
Yeah, look into it.
There are no further questions. Thank you for listening. I'd like to thank our keynote speaker again, and we are going to have a break now. We have our vendor exhibits outside. I would highly appreciate if you could meet with them as well. And if you could be back here a little before 1:30 p.m. And our afternoon, we have some great talk with reference to the children and outreach programs and embryology. And see you back at 1:30. The lunch is at the lunch. Thank you.